I think most of us are already in. So we want to start with the first session. It's on crop production and improvement. And we will start with our keynote speaker. Under that theme is Professor Miriam Kinyua. She's already here, and we welcome her to give us her talk. Yeah, but briefly, Professor Kinyua is actively involved in teaching, extension, and research in agriculture, crop improvement, and biotechnology at the Department of Biotechnology at the University of Eldoret. Yeah, Professor Kinyua holds a PhD in plant breeding. She also has an MSc in plant breeding and genetics, and a BSc in agriculture. Yeah, all her degrees were obtained from the University of Nairobi. She has, as an active scientist, she has won over 20 research grants, and she has used this to support many postgrad students who have gone on to graduate and move into diverse fields. She also has a prestigious award of the Order of Grand Warrior. This is a prestigious national award, and she won this from the president for her efforts in agricultural production, especially through research, and activity in women science networks. And she's very instrumental in mutation breeding. She's an expert in mutation breeding, and she uses this technology to improve crops. And she's supported in this technology by FAO and the International Atomic Energy Agency, who have continued to support her research work because of the excellence that she also brings on board. So she has used this expertise and these resources to develop many varieties, crop varieties, especially potato. And because of this, she has also patented many varieties, some of which are called Eldo Mavuno, Eldo Katie Black, Eldo Katie Cream, and these are very popular varieties among Kenyan farmers. So Professor Kinyua has done a lot in agricultural production in the country, and because of her expertise, we just want to listen to her talk. So Professor Kinyua, feel welcome.
Ladies and gentlemen, good morning. I'm happy to... I'm going to talk about genomics as a complementary wheel in accelerating crop improvement and production in Africa. In other words, genomics is not the only one, but it needs to move along with others. Before I can get into the science of it, I thought it's good for us to look at what is the food demand in Africa. And to understand that, we need to look at the realities of food security and production in Africa. And these realities include no production means or hardly any. Poor production means, like inputs, we don't have, varieties are not there. They are not good enough. mobility is poor, and the population is increasing. So all those are realities in food. On the other hand, and pulling in the opposite direction is the global macroeconomics. The realities of macroeconomics globally. What's our GDP in the different countries? What are our socioeconomics that affect production? That is pulling in the opposite direction, and it's not in the hands of the African common farmers, consumers, and processors. It's in the hands of the global setup. So the two are pulling against each other in this situation. I looked through, and it is reckoned that Africa has got a population of 2.4 billion. Of that, 20% sleep hungry. I mean, they do not have food that they can talk about. So they are facing hunger. Imagine more than half of the hungry people are in Africa. While actually, we occupy 20%. 20% of the globe is Africa. But more than half of the hungry people are in Africa. So you can just imagine how grave that is. So what are the challenges? Why shouldn't we have these problems? From my review, it came out that climate change and ecosystems is one of the major problems that cause food insecurity and poor production. We live in the tropics. There's the sub-Sahara area. A quarter of Africa is covered by the Sahara system. The landscapes are all contributing to the poor production and the other challenges. What about the infrastructure systems? You have a lot of production in some areas, but they can't move that food. It can't be taken to the other places. Then you have storage facilities. There are none where this food is being produced. So it can't even wait to be transported when the system is better. So you can see that apart from the ecosystem and the climate change, the infrastructure itself, and if you think of infrastructure, don't just think of roads. It's in its entirety. It's compromised, and therefore it causes problems. Available arable land, I've just said that the Sahara coverage is about a quarter of the whole of the continent. Then we have the tropical forests. Then we have the waterways and the water bodies. So when you think of all those, they take up lots of that is being accounted for Africa, but actually it is not arable. It's there, but you need a lot for it to be used. Now, with that background, today we are talking about genomics. And I have counted it to be one of the tools that can be used in production. And since me, I'm a crop scientist, I will more be biased on crops, but that doesn't mean that it is only crops that can be let's see. Hi. Yeah. It's not only crops, even livestock are included there, but I will be biased on crops because crop science. So you find that if we talk about crop improvement, we have the conventional one that we know about, hybridization, then selection. We have genetic engineering, which caught fire and now it's cooling. We have biotechnological tools like micropropagation. It's also part of food production and improvement. But genomics has got a part to play. And I thought I should be able to bring out which aspects of genomics would play into crop production and improvement. Characterization has been made easy because we've understood genomics. It is now possible for us to characterize the whole genome. Before, we want to talk about phenotypic characterization and genotypic characterization, but now we genetic characterization, but now we can talk about the whole genome. Apart from that, we have marker development. Now we are talking about markers. SNPs are being used for us to be able to characterize, to get to know where the gene is, to get to know the alleles, the differences. So it's really becoming quite a transformation. On top of that is the selection precision. When we were in school, we used to estimate genetic value because we couldn't be able to see it. We want to see the phenotype, and then we want to be able to calculate the genetic value. Now, selection is becoming much easier. We also have the speed of selection. Conventional, you go 7 generations, that's the time that you'll be saying, I have enough homogeneity for me to go on to make a variety. Now, early selection for the early populations, you can be able to select in even F2 or M2 or S2, whichever class of science that you are applying. Why? Because you are able to look through and see the genome as is because of the genomic tools. Okay, now I've just been talking about genomics, and maybe some of us haven't fathomed what genomics is. So by definition, genomics is the study of all the genes and how they interact between themselves and also how they interact with the environment. So the three are key, that it is interaction between the genes, they are the genetic pathways, they are the physiological pathways and all that. You are able to see how the genes are interacting between themselves, how they are being affected by the environment, the environmental factors. So that is what genomics is. There's the link between the genotype and the phenotype, which we were not able to understand. But now we can understand it through genomics, and it has really transformed crop improvement. It enables faster gene discovery, and therefore, of course, we can do precision breeding instead of the way we were just doing breeding by imagining we understand the inside of it just because we can see the phenotype. But there was a lot that we were not able to see. Now we can be able to precisely identify the genes or the alleles that are associated with the important traits. And that helps us to have reduced breeding cycle and also to deploy sequencing and genotyping. So what is the importance of genomics? We have talked about gene identification and characterization, where we can do it precisely, and also we can be able to study genes responsible to the traits, we can be able to do marker development, which helps us in the selection. And we are able to understand gene expression when under stress. And in this case, stress is either biotic or abiotic. Abiotic stress, like drought, salt, acidity, and all that, or biotic, where you have the diseases and the pests. So now we can be able to understand the gene expression. We also can be able to understand the pathways. Through genomics, we're able to understand the stress response pathways. And when, of course, you understand the stress response pathways, then you can be able to do improvement or even production better because you have the understanding. How can we apply genomics? We can be able to apply genomics to be able to handle complex traits. And then we can use genome-wide markers. When you use genome-wide markers, before we were using genetic markers or even phenotypic markers, but now we can be able to have markers which describe the genome in its entirety. And this, of course, helps us to have faster and more informed selection and also to make decisions. We can be able now to look at complex traits. Before, you had to estimate or to imagine or to be able to deduce, but now you can be able to see it. Other applications is prediction of genetic merit, because before you would run with material thinking, oh, this is very good material, and when you are 3 generations down the line, you realize it has traits which you do not like, or it has traits that the farmers do not like, or it has traits which the consumers do not like. And for you to go back to develop new material, it will take you another 7 years. But now you can be able to have to know the genetic merit of the material through genomic estimated breeding value. Once you know the genotype, then you are able to estimate its breeding value. We can also simultaneously leverage information from across the entire genome. You're able to look at the entire genome and be able to do simultaneous information handling, and the genome-wide markers are helping us in a great deal in this. When you are able to estimate the breeding value without having to go to the field, great. I mean, it's a great thing. You're able to know this material of mine, this is its value. And you've only done it in the lab. You haven't yet gone out to the field. Of course, at the end of the day, you need to go out to the field for adaptability and all that. But at least even as you go out, you'll be able to know what kind of interaction this genome will have with the environmental factors. The story sounds very good. It's like it's butter and using a knife, but of course there are challenges in the application of genomic tools. Okay, so the cost is there, but of course the return to investment is also there. I mean, you get high return to investment because of saving on the seasons and also being able to predict, to do the estimation more clearly. But of course, the cost is there. And of course, if the cost is there for Africa, you can imagine what challenge it is. For you to be able to do genomics, you must have genomic data, and this might be missing for the crop that you wish to work on, or it might be limited, and therefore that can be a challenge. Some crops have got high genetic diversity and complexity. It's not a linear thing. So when it is like that, then it can be quite a challenge. Then the genome structure, if you are working on a crop that has got a complex genome structure, then of course that becomes a challenge because it will hinder the precision in genotyping tools. Other challenges include this translation of genomic data into efficient and applicable improvement program. Because just having data is not the final. You need to be able to interpret that data, and it needs to become applicable. So, of course, that can be a challenge if you do not have that ability. Quality reference genomes, because of course you need to have a reference genome. So if you do not have one, or it is not of high quality, then it becomes a problem. There is innovative breeding approaches. If you do not have them, if you do not have biotechnological tools that go along with the genomics, or if you do not have other breeding tools, then it becomes a problem. And I've written there the issue of marker assistant selection and marker back crossing, and also genome selection. So if you do not have those tools, then of course, even the genotypic tools do not become useful for you. So therefore, now after all that story, where does genomics, which is the place for genomics in Africa? I thought of two classes of stakeholders who are very high on the rank. And the highest one, I felt, was the farmers. As much as it's us scientists who will do the genomics, we are not doing it for just ourselves. We are doing it for the farmers. And this happens within the breeding programs. We are doing it for the farmer. And one of the very useful strategy is having farmer participatory breeding. But you can imagine you call a farmer and you're trying to show them all those genomic bioinformatics. That is not applicable for them. They won't even understand. So, but at the end of the day, it is very key for them. They are the key stakeholders. The agro industries, where quality is an issue, you develop a variety. Does it have the industrial qualities that will be useful in processing or in even packaging and all that? And, can it be value added? Is it possible? So those are, I felt that with the agro industry, you need to think about the quality as well as value addition. And can you have farmer agro-industry participation when you're developing the varieties, or even when you are producing those varieties? My key or my thinking or my way of looking at it is that there must be tailor-made genomic application for it to be useful in Africa. It must be compliant and friendly, and that is of necessity even as we do we apply genomics in Africa. Genomics can be applied for variety development, where having the information, the genomic information, helps you to make better varieties. apart from varieties, you can also be able to come up with breeding lines, which now you can use using other methods like mutation or conventional or even genetic engineering. You can use the breeding lines, not necessarily to get to varieties, but to be used in the scientific knowledge. And genomics also can be used for us to just understand, having the information, being able to understand what a variety is made of or what a crop requires, just because we've been able to understand it. So with all that, I can conclude with the following points. That genomic information should be integrated into growth and resilience of food production. In other words, genomics alone, having the genomic understanding, does not help us if we are not able to integrate that information into growth and resilience of food production. And the other conclusion point is that sustainable food security and economic growth requires complementing tools. And that's where now the genomics find a place, that it isn't a replacement, it is a complement. The baseline is that we need good breeding designs, because if you don't have a good breeding design, I mean, then it doesn't matter how much genomics that you know, or even how well equipped you are. You still need a good breeding design for you to be able to make use of genomics. The other very key point that I felt I should conclude with is that you can not live alone. We need partnerships and collaborations if we want to progress. And that's why we must think global. We can't just think African and think that we have reached. Partnership are key, and collaborations. Next generation sequencing, unless we get to understand that, then we will not be able to apply our genomics well. And the other point is that without bioinformatics, then genomics alone doesn't help us. So we need to be able to interpret the data, and we also need to be able to understand the information that we are getting from the genomics tool. So that's why it is very important to have bioinformatics. Application of SNP data for candidate genes and biological mechanisms, it's the way to go. We need to be able to get that SNP data well. I mean, it needs to be clear, and it needs to be applicable, so that we can be able to know the biological mechanisms, as well as which are these traits that we are interested in. Yeah, so with all that, and thanking you for listening, I wish to pause. Thank you very much. Yesterday I was hearing people being asked, are you okay to stand there? And I am okay to stand here.
Yeah, thank you, Prof. And let's applaud Prof in a round of applause. Thank you for the informative presentation. Now I'll welcome any question. I'm Dr. Vivian Tue. I'm coming in to chair this session. Yeah. Any question, any comment, it's welcome.
Yeah, thank you for the interesting contribution. And I'd like to take up what you mentioned about the necessity for reference genomes, high quality. And am I correct in assuming that the crops are used, say, for example, in Kenya, are often deviant from those used in Europe, for example. So it is really an issue that these reference genomes are not available at all. Is that correct?
Yeah. Okay. Very important question. You see, we have been imagining that just because it's a wheat crop, it's a wheat crop. But we've already described genomics as the interaction of the genes with the environmental factors. So rightly to say is that if a crop has grown in Europe, that's how it has formed itself. I mean, its entirety, its genome has got the package of the environment in it, and therefore it's going to be different. In fact, when we were doing potato, and we are like, Netherlands has always been growing potatoes, and Europe, so maybe we can get their potatoes as the reference point. And you realize that the information that comes out is so different. We were also doing the black bean, I mean, Dolichos. It's now this is called lab. And you think, okay, maybe I can use the cowpea as the reference genome. And you get to realize that they are so different. So to answer the question is that there are differences, and that's why getting quality reference genome is one of the challenges. But of course, it's not limiting to the point where it's undoable. So we must face that challenge and make it as an opportunity. Yeah, thank you.
Okay, any other question? Comments? Oh.
Yeah, so thank you, Prof, for that nice presentation. Mine is just some views. There is some fairly substantial misinformation or understanding of these technologies, especially in our country, Kenya. Yeah, anything that is improved from the lab is generally viewed as GMO. Yeah, so a few years ago, you remember the uproar, the public debate, yeah, when there was mention of this GMO maize. Yeah, there was a suggestion that the country could allow importation of GMO maize or GMO foods into the country, and there was a big debate over that. So during that debate, I saw that there was a lot of misinformation. Anything that is improved, people view it as GMO, and so they generally begin to have a negative attitude, yeah, towards these technologies. And if that misinformation or misunderstanding becomes widespread, then it's likely to impact, yeah, the effort, yeah, the scientific effort to produce these varieties, because they are very important. Yeah, and also these things, this misinformation appears more common in plants or crops, perhaps because crops or plants have been improved, yeah, to a larger extent. So I don't know what you would comment on this, and perhaps what effort do you think we can apply, yeah, towards, yeah, making the population understand that not every improved variety is GMO, and therefore something to shy off from. Thank you.
Yeah. Thanks, Daktari. Maybe I should have included that as one of the challenges. I must have missed it by a whisker, because misinformation and lack of awareness is one of the greatest challenges when it comes to application of scientific findings that are realistic. But because there is, first of all, misinformation, those ones went ahead of you and they told stories, now it becomes a challenge. Misinformation can be either out of ignorance, that actually they do not know and they think they know, ignorance. But it could also be just to have the other side of thinking. I would say it's like it's a competition. There's competition. I mean, scientists will have what we used to call schools of thought. And just because I'm in a different school of thought, I want to spoil for the other school. You see? Not because I'm ignorant. I'm just being arrogant. There's also the facts. Facts can also make people to want to be careful. And Dr. I would wish to say that misinformation can even be within the scientific group where I think I know your science. I know about fish. I was I was telling Prof that why are you calling them weakly electrifying? I mean, I thought, and he took more than an hour to tell us about weakly electrifying, and I'm like, how does a fish become electrifying? You get? So in that case, I am ignorant. And if I go to tell about his story, I will be telling the story the wrong way, not because I have any bad intention. It's because I do not have the right information. So I would comment by saying that we shouldn't tire. We should just be ready to create awareness, work out, have strategies of managing the misinformation. Let me tell you one experience I had in the field when I started applying mutation techniques. My first wheat variety, it was beautiful, it was outstanding. So farmer participatory, I took them to the field. Twendeni mkaone. Twendeni mkaone is, let's go, you see this beautiful thing that I have come up with. And the farmers were so excited, and they were going into the field and saying, beautiful, it has all the characteristics that we need. I mentioned that this is through irradiation. Everybody left the field. You know, they just walked out, and I was like, but you've already been irradiated if there's any irradiation that was taking place. Why? Because they thought if I am using irradiation to develop the variety, the material is radioactive. So we walked out, we stood out, and I created the awareness for them. What is application of mutation techniques? I gave them a lecture, and they were like, okay, and they took the variety, and the variety lived on. But if I didn't have the gut to tell them that, I would have just said, ignorant farmers, you can get away. They are the ones to use the variety. So colleagues, we just need to be not feel put off just because someone is unaware of what you are talking about. Let us have strategies of creating awareness and removing the misinformation so that people can be able to appreciate the science of it. I might be holding a different opinion about genetic engineering, but that doesn't stop my colleagues who want to have genetic engineering as their line of crop improvement. So we just need to be able to help each other to understand each other and the public and the other stakeholders and government. By the way, don't think that the policymakers understand some of this scientific jargon that we have. As much as they went to school with us, they didn't go to school with us to learn the science. They went to school with us and they took a different line, policymakers. They are doing economics and all that. So we should be ready to help them to see the right thing. When I make the presentation on mutation breeding, my own lecturer, the one who had lectured me, asked me a question, and I was like, so even my own lecturer does not understand mutation techniques. You know, why? It's because me, after school, I was able to be exposed to application of mutation techniques, now not as a theory, but as a practical. But him, he was doing it as theory and read it in the books. So members, let's just be ready to have strategies to create awareness and to help people to have the basic understanding of what we are talking about.
Any other question? Okay. Maybe the last.
Sure. She's a keynote speaker. We need to explore a lot from her. Thank you, Prof, for the wonderful presentation. I have two comments or maybe inquiries. One, maybe I need you to shed some light on biotechnology versus GMOs. What is the difference and then the similarity? Where is the link between the two? Then two, I would like to know if there is any platform where breeders meet these guys in the industry that are producing the agrochemicals, so that at some point maybe you can have can harmonize on common issues to avoid a lot of these chemicals flooding into the agricultural land. And also, of course, I see breeders and these people in the chemical industry, you almost look to find the same solution towards crop challenges. Yeah. So where is the link? Thank you.
Okay, two challenging questions, but good ones. Let me do the easier one, because it's not social. This is science, biotechnology versus GMO. Genetic engineering is one arm, one branch of biotechnology. Biotechnology is broad, where you have micropropagation, like tissue culture, and yeah. So that is one arm of biotechnology. Then you have diagnostics, where you use biotechnological techniques to study, to diagnose the relationship between the pest or the stress and the crop. So that is another arm of biotechnology, diagnostics. Then now genetic engineering that produces GMOs is another arm of biotechnology. And finally, there is the marker assisted selection, where you use markers to, like the way I was talking about SNPs, then you have AFLPs, rapids. That is also another arm of biotechnology. So biotechnology is broader, and it encompasses genetic engineering that produces genetically modified organisms. Genetically modified organisms is when you take a foreign gene and you insert it into the genotype, into the genetic makeup of an organism. So it must be a foreign gene for it to qualify to be genetic engineering. So you see, like some will take, like now if I wanted my plant to be electrifying, I just go and get the gene, the fish gene, and put it into the plant. Then, people want to do aesthetics of plants by getting the gene of the glowworm, so that their plant can also glow. So that is getting genes from different organisms and bringing them into a non-related organism. That is the genetic engineering. So it's one arm of biotechnology. Harmony of chemicals and breeders. I ask myself the same question. Like I'll say, I'm breeding for disease resistance. The chemical company wants to bring a chemical that is going to kill the disease or that will going to help the plant to be So basically, we have the same goal, but we are using different lines, and we never agree. I mean, if someone realizes that I'm going to develop a variety that is resistant so that their chemical cannot be sold to control the disease, why would they be my friend? You see, I think what we need to do is to be able to think about the consumer. I mean, think about the goal, and then we'll be able to come up, because I can't breed for everything, and the chemical company can't produce everything, but we should be able to look at the middle ground where it is beneficial to the clientele, which we don't do. And I can imagine us having a meeting between breeders and the chemical company, AAK and Plant Breeders Association of Kenya. I think it's almost impossible. But it is the way to go. It is the right way to go. Maybe now that we've realized that we can't be Mr. All, we might want to speak to each other. It might force us to get to that. Yesterday I asked Paul the question of, are you sure that we won't rewrite science now that we've got genomic tools which are informing us better things than we thought? I think we should be ready to change our minds. And science is growing, it's always changing because of the understanding that we have. So I'm with you, but bringing us together, that third party, that third person, the go-between who is going to bring us together, they really need to work, because we have different motives. Actually, it's our motives that are different. Our goal is the same, but our motives are different. I don't know whether I have a... You can, I mean, I don't know what you would say, because we are discussing. It's a discussion. Maybe, Prof, you need to give him the... Yes, give him a second chance, because I can see you just...
Yeah, thank you so much. Surely, I see the other guys in chemicals going into business.
Yeah.
And that compromises the health of the consumers. Then breeders are looking at environmental probably harmonization and reducing on the toxicity of these chemicals that we use. I am looking at Pelham. I've had it in Uganda, I've had it in Kenya, and the other time I attended one of their webinars. They were talking about signing, were they petitions on reducing the chemical usage, or maybe totally banning these chemicals. But the issue is, fine, we ban them. What is the solution? Are we Mr. All, just like you said? So can we find a common ground that surely we all come on table, so that even when they are in business, there are some limits on where they can surely reach, so that business shouldn't override everything. Likewise, as we see, how do these other players come in to sort this issue? So it is over to you, Prof, and the platforms that you have to advocate for this. Thank you.
Yeah, very true. I mean, if I was government, I would lock the chemical companies and the breeders into one room and tell them that I want a harmonized way of managing, because if I am in government, it means that I've been given the population to take care of it. I would just say, and since they have the basic understanding, some of these things, I would just say you must give me a solution that is caring about the consumer. I would say that. But as I stand on this podium, there are those things I can't dare say. I mean, if I was seated there where you're seated, I would ask it as a question. But now for me, if I dare say, it will be Miriam said. And so that is what you think, or who sent you, you know, because there are those things that even you know very well that we can be able to solve them, but we are not intent on solving them. We are either intent on making business, or we are intent in remaining in science and being known that we are senior scientists, or, you know, the intention is what we are wrong in. And we can try. Daktari, maybe you can, because you are neither a breeder nor a chemical company, you can initiate another conference, and we sit together. And this shouldn't even be global. It shouldn't be countrywide, because you see, why did we develop the products that now we are saying that we are banning? And when you are asking actually a genuine question, when we ban them, what is the alternative? You see, we need to have an alternative that is workable. So it's a complex thing, but if we get serious, we can be able to come up with a good solution.
Thank you, Prof. Unless there is a burning question. Okay, if not. Oh, okay. Now the last.
Thank you very much. Not actually a question, but maybe just allow me. I'm not supposed to ask a question. In reference to this theme, from the era of genomics era, and African rush towards sequencing its genome for the sake of increasing or conserving our agrobiodiversity. It's very funny. I'm sorry, Prof. You taught me this.
Yes, I did. Let's see what you're going to say.
Professor Anne Mugai and African Plant Germination Project is really working together with other teams to ensuring that Africa meets the standards or matches the rest of the world towards genome sequencing. And one of the purposes is biodiversity conservation. Because of intensity of the breeding, there's genetic erosion. We are losing our biodiversity, where is the source of our novel genes for breeding. So I think in this regard, and with respect to our discussion now, we are looking forward to at least how do we conserve our biodiversity with regards to our breeding. Because once we lose it, we shall lose our food. Everything is improved. Be it animals, be it all the crops today, be the little regard to the ancient ancestors where we owe our source germplasm. So I think that could be the nexus. Suppose we lose the genomes of the ancient for our breeding materials. What next? What if everything we talk about today is improved? And therefore, what is our position in the next breeding circles? Thank you very much.
Yeah, maybe Naman, you shouldn't have told them that I tell you that once you've made the change, it is not reversible. You know, like the way we say reverse genetics. You can't reverse and return it to what it was once you've made the change. Once you've done the gene disruption, then it's not possible to repair it so that you get back to the old one. It's a good concern. We've already shown how Africa is challenged in the ability, and the collaborators that you will call along, it's just like the chemical company versus the breeders, or the rat versus the mouse, or the, it's comparisons. So if let's say you called me to come and help you genotype, characterize your material for the sake of conservation, and then when I assist you, you will keep it to yourself, or you sell it to me so expensively, and you do not have the ability to do it. You know, those are the kind of situations. And that's why I showed my second slide was the pool of macroeconomics, global macroeconomics, and the need, the realities of the food improvement and production. So, Naman, I would say that those are realities, and it's up to us to decide how to move and also look at the fact that we still need to feed the increasing population. So we must align all the realities and be able to carry along with that. Genetic erosion, of course, it's happening, and happening very, very fast. We've awoken when we've lost many of them. And that was like the first question I was asked about the genome and the differences. You know, you might be thinking that you're keeping that one that was there in 1900. Well, actually, you lost it a long time ago. It looks the same, but it isn't actually the same one. So, yeah, we have a lot. We have to prioritize the things that we are going to do. And we also have to remember that we are working with collaborators and funding agents and partners. I hope I've answered your question, Naman, by hiding behind the actual words that I should use. Read between the lines.
Yeah. Thank you, Prof. And I think we still have time to engage with Prof in our breaks. Thank you, Prof, for all the responses, for your presentation, remarkable plant breeding successes, and fast experience. So let's applaud her lastly, and thank you. We have about 5 presentations for this session. I think we'll have 1 presentation and then we break for tea. So I'll call upon so the next presentation will be online. We'll have 1 before we go for break. So this will be by Ahmadine. I hope I'm pronouncing it well. The talk will be on unveiling evolutionary genomics of tree crops across continents, harnessing adaptive potential for food security in a changing climate. Is it Ahmadine? Ahmadine. Are you there? Just a minute. Sorry. Yeah, Ahmadine, I hope you can hear me. Yes. Yeah. So welcome to the symposium. We are ready to listen to your talk. You'll have roughly 15 minutes and maybe 5 minutes of engagement. Welcome very much.
Yeah. Thank you so much. So thanks a lot, and I missed to not be here, and for that I would have loved to see you all. Actually, it was a way for me to meet you, but finally I have to postpone my venue in Kenya next year, so I will be there next year. So today my presentation will be actually mostly on the apple tree, the work we have done over the past 10 years. And the aim of this presentation is to show you what we would like to do in a model system in Eastern Africa. Actually, I was in Kenya in September, and I was planning to come in Tanzania in 2 days. Before, but I could not make it this year, but I will be there next year. So the aim of this talk is to enhance collaboration. So if you want to reach me out after, please feel free. So actually, I'm working on fruit trees, and you will see that I'm moving toward the study of trees, and based at New York University in Abu Dhabi in the UAE. And I have been working for a long time in Paris at CNRS. And actually, I'm focusing on trees because they are key species in ecosystem and agrosystems everywhere in the world. And the diversity in Africa is astonishing. In the Middle East as well, and Eastern Africa. And that's why I moved to Abu Dhabi to focus on this diversity. But before, I was working mostly on temperate species, and especially on the apple tree. I'm going to talk about that today, but also other trees you are seeing here. And surprisingly, we still don't know the responses to climate changes. And one way to answer this question is to study both the crops and their wild relatives and their domestication history. So we have done that using microsatellite markers long time ago on the apple tree, and actually maybe you eat one apple a day. As we say, one apple a day leaves the doctor away. And we have seen that this apple come from Central Asia, from a wild apple species, and then was spread across Eurasia. And another species contributed to the cultivated apple genome by crop to wild to crop integrations. So it's a typical process of domestication followed by diversification. And then we went in Iran with Ahmed and Hamid, and we have also seen that Malus orientalis, another species, contributed by wild to crop integrations. And later on, when Shillong came into the group, we have seen that another species, Malus baccata, did contributed to the cultivated apple genomes. And so we used a different kind of marker, from microsatellite to SNP marker, and we have seen actually that there is a complex domestication processes involving several right species. So that was a very first start for us, and it took some years to see that. And the next question was, which genes are involved during domestication? And so this was the work of Shillong. We wanted to understand how many and which genes were involved during domestication, and especially what is the genomic landscape of this wild to crop integration, so from the wild to the crop, and what is the genomic architecture, and which genes are involved, or they're climate related or resistance related. So we use SNP markers, and we looked at different kind of selection in the genome of both cider and dessert. You may know that in the apple, you have apples that are used for making pie, and the others that are done for making cider, so the beverage. And so we expected different signature of selection and selective pressure during the domestication, and we can split them between hard and soft sweep. I won't enter into detail, but basically what we have seen in the dessert apple, so in the apples that you probably have eaten one day in your life, we have seen several genes that you see the name here under selection, positive, so it could be hard and soft sweep, and they were related to fruit length and diameter. On the other side, we have also seen some genes related to fruit quality and resistance to pathogen in the cider. What was interesting is that you had different footprints, sometimes soft selection, sometimes hard selection, depending on whether you were a cider or a dessert, which suggests that you have different evolutionary dynamics during the domestication process. So we're okay, it's interesting, and we have seen massive hybridization before constructing the domestication history of the apple. So in those genes that we have seen under positive selection and that we have validated using differential expression analysis, do you have overlap with introgress region? So that was our next question. And then what we have looked at is the haplotype structure. I will make it short, but here you see the different color are the population we have detected. So the cider apple, Domestica cider, and then you have all the wild relative, Sieversiae, the Central Asian, and then the European. Here you see basically all the individual we have sequenced, and each line here, it's a haplotype. And what you see here is that all our cider apple here, so haplotypes, they are actually clustered with all the wide haplotype of the European apple here, suggesting that you have adaptive integration because they are targeting in sequence here that are associated to texture association losses, so the texture of the apple. So you have overlap between region we have seen under selection and region we see in progress. So we have seen also that in the dessert apple, so some genes related to the phenolic content, which are important in the apple because they are related to the taste and the variety and the diversity you can see in apple tree. And we see also that dessert apple haplotype actually are embedded in the wide apple gene pool. So this is very interesting for us that we see that wide apples are a major source of adaptive genomic variation for breeding program. And we wanted to dig further in this wide apple genetic diversity. So we use microsatellite markers first to look at the genetic diversity of the European wide apple across its distribution. And we did see different genetic groups. So in Europe, 2 in France, in green and yellow, 1 in blue, here in Scandinavia, another one in Eastern Europe, in red, and then in Italy. And using simulation called approximate Bayesian computation combined with coalescent-based simulation, we have seen that this population resulted from the past glacial maximum change of temperature. So 20,000 years ago, most of the temperature in the world decreased, and especially in Europe, you had migration of temperate species into the so-called glacial refugia, so Spain, Italy, and the Balkans. And so population did differentiate. And then 10,000 years ago, during the Holocene, all the population migrated back to the north. And then you had hybridization zone and suture zone, and they recolonized the northern part of Europe. So this was very interesting result because we see that the European wide apple responded to climate changes, and it opened a lot of questions regarding whether you have genetic adaptation of the species to climate, or do you have plasticity, which are key questions when you are also looking at the cultivated species to do a breeding program, and which genes allele and even EP alleles are involved. So to answer this question, we went back on the field. We went in Europe, sampling different population of wide apples. We love to do field work, and we are starting to do that in Eastern Africa. And we plan to continue that. I'm looking for collaboration, not on apple. I will tell you a bit more at the end of my presentation. And so we did sample a lot of wide apple seed, and we grew this seed into a common garden, a wide apple orchard. And I put all this logo because it has been an adventure when I was at CNRS, and we set up this orchard with a 5 population and planted about 500 orchards. The aim of this orchard are for us an experimental station to test for fruit tree adaptation to climate. It's also a source for apple science, for conservation and breeding programs. And we deployed this system across Europe. We did replicate in Europe, so you can see here in yellow different wide apple orchard we have set up. And we have also cultivated apple orchard here in red in Belgium, France, and Spain. One is overlapping in Angers. Which is very well renowned for its horticultural competency expertise. And so the aim of those orchards are to understand the responses of wild and cultivated apple to climate. But as you can imagine, it's a long-term experiment. So we have actually tested that in control conditions. So here it's just brief results based on flowering time and photosynthetic activity measured on Malus domestica, so the cultivated apple on 2 sites in Spain and France. And what you see is that you have a phenotypic differentiation so far between sites, and I'm not spoiling the next result, but Maxime and Mathieu are working on it. And so, as I just told you, we have done beyond this in natura experiment, control condition experiment in climate simulators called Ecotron, in which we did use some seed sample in Europe and we put them in their respective climates and also their transclimate. So we did transplantation to understand whether you had local adaptation or plasticity in the wild apple. So we did measure the phenotypes, so physiology, morphology, performance of the seedling, but at the same time, we also look at the gene expression and the sequence genomes. And I'm not going to explain all the results, but basically it's very interesting because we see very high plasticity of the wild apple, but also pattern of local adaptation to climate. And we see the very high stability of the cultivated apple, whatever the climate. And we also have detected some core genes related to climate adaptation in the cultivated apples that could be used for breeding program. So this is under revision in a journal. And so the next step is using the field experiment I show you here, and especially the field data, to predict the response of this crop and wild apples to climate changes. And we are not only doing that in the apple tree, but also in almond, peach, apricot, grape, and wine, using genomic vulnerability analysis, combining SNP data and climatic data to estimate to which extent each cultivar can be fit to the changing climates in 50 years, more or less. So we are developing a pipeline, a fair pipeline that will release soon, and also that we want to apply to the species we are going to start working in Eastern Africa. And we are starting also to look at epigenetic marks. As you may know, some regions of the genome, especially transposable elements, are methylated or not, especially in plants. And we believe they play a key role in the response of plants to climate changes, and especially of trees. And I will explain you why after. And to look at methylation, you also need to look at transposable elements in the genome. So we start to look at transposon insertion polymorphism, especially in the apple tree. So this is the work of Soumya and Aurélie, but also in other species I will present you at the end of the presentation. And of course, for that, we also need pangenome, not only to look at transposable element, but also other structural variants. And so that is the work which is ongoing. And we have also looked at methylation marks using long-read sequencing and also using enzymatic EM-seq, basically. And we do that with one of my colleagues in New York, NYU New York, and with Anthony, with the lab manager in the team. So to sum up this presentation, it's we have seen a very complex domestication history involving wide contributor. We see that these contributors have provided key adaptation, key variation to the cultivated apple gene pool through wide to crop integration. And we are using a multidisciplinary framework to understand the response of apple, both wide and cultivated, to climate changes. We have seen that you have adaptation and plasticity in control condition. I invite you to go to the preprint of these results. And we are also looking at that in natural conditions. And especially we are seeing genes involving climate responses. And also we are looking at epiallies and especially methylation and transposon insertion that can play a role in the adaptation to climate changes of this long-lived perennial. And why we are looking at that? Because we believe that actually methylation and epigenomic variation, it's a flexible way to respond to climate change for long-lived perennial, which are planted in orchard for years, sometimes centuries, depending on the species. And actually, just as a take-home message, so genetic, epigenetic, and ecological diversity matter. We talk a lot about GMO, but I also believe that going back in the wild, harnessing wild diversity could be also very interesting. And biodiversity, not only the wild-related, but also agroforestry program to favor biodiversity insight can also help to face climate changes. And so just actually why I was planning to come in Eldoret, but I will come in any case soon. It's actually all these questions, we are going to apply them to other crops and not only fruit trees. So we are developing projects on figs. As you can imagine, figs are a keystone species in ecosystem and agrosystems. And we're also developing on acacia and mangrove. So these two species are also key species in the Middle East, in Eastern Asia, and also in Africa, and they are co-evolving with microbiomes, and figs are co-evolving with wasps. So I'm very fond of co-evolution in the case, but they are also very good model to better understand the impact of climate on diversity. And so I'm here, not really physically, but soon I will be, to do collaboration to generate epigenomic, genomics data, and ecological data and resources in a fair mindset and long-term program for agroforestry, like fig tree are used in agroforestry, vachelia as well, and mangrove tree are used not in agroforestry, but there are plantation program in the UAE. Actually, they are big program, but I'm thinking to do collaboration on this topic also with collaborator in Eastern Africa. And the idea would be to better understand how crop, and especially perennial crops, are resilient to climate changes and parasite outbreak. I haven't talked too much about parasite, but we have program on parasite adaptation and local adaptation of crops as well. This is for food security, but we have also program for against desertification. So on that, I really want to thank you for your attention, and I want to thank all the founders of these programs, all the people that has been involved in all the programs since a lot of years. Of course, my colleague at NYU Abu Dhabi and NYU New York. And also, last but not least, the team here that help every day on this work. And please feel free to contact me, and I will also probably contact you by email. I've seen the amazing program of your conference, and I would have loved to be there. So I will be happy to answer your question. I want also to thank the organizer for the conference. Thanks a lot.
Applause. And welcome to East Africa or Kenya for further research. Yeah. So we'll welcome a few questions or comments. Yes.
Yeah, thank you. I'm Ralf Tiedemann from Germany. Very interesting presentation. I come back to your. It was in the beginning of your talk where you inferred selection from your genomic resources, and my question is as follows. I mean, typically in domestication, I would argue from the phenotypic level, if you do optimized breeding, it should be by definition hard selection, right? You just take those which show your preferred trait, and you only propagate them. And my question is, the selection you observe, is that always related to the traits the breeders have looked for, or is it in a sense unconscious selection to the environment, which people were perhaps not even aware of it happened, but it simply happens because those better survive than others?
Yeah, indeed. Actually, what is expecting, like when it's like a trait that has been selected by a breeder. I don't know if you hear me because I have a bit problem of connection, but basically you expect soft selection when it's unconscious, and also when it's a trait which is not like a qualitative trait. So we observe most of selection, for instance, in cider, because in cider, we don't select like a very good apple, a very well-shaped apple. We are going to select actually trait that it's a population that we select. It's a diversity that we mix together. So the selection is less strong than in the dessert apple. So indeed, breeder targets very specific traits. So you expect hard selection, but actually for some trait, and especially resistance, we can expect soft selection. And you have also two paces of selection. You have the one that has occurred during domestication, and then the ones that occur during recent breeding. And so the ones that occur during recent breeding could be like the very recent one, so very hard sweep. And the one which is much older could be also soft sweep. And especially in trees, we expect a lot of soft selection because you have a lot of standing genetic variation. So that's really depend on your model. In annual, you expect probably more hard selection. But it has to be validated, to be honest, because soft selection is not so easy to see. And the tests that exist are not so numerous for the moment. I don't know if I answered to your question. Well.
Okay, one more.
Thank you very much, Amadine, for that beautiful presentation. I think the subject of climate change and the issues of resilience, I think trees, fruit trees, is an important area to be able to come in to address some of these challenges. So my question has to do with you really have an interest in trying to address this challenge. And when you come to like Africa, Kenya, for example, the apple, we have few apples. And there is opportunity to do apple farming in Kenya. And considering that this is not an environment that apples have been known to grow. So we have good apples, bread improved in Europe and other temperate countries. How can you be able to now actually utilize this to improve local? Because the demand for apples is huge in Africa, and the benefit of this in terms of climate resilience would be something that if this collaboration can be able to bring, especially to the farmers, and this is big industry in terms of the economics. If we can be able to produce this would be something very beautiful, very good for the farmers, and we would really love to see how probably the knowledge from the rest of the world can be able to benefit our farmers. Thank you for that. That is my comment.
At the same time, so actually you are opening a very interesting topic for me, because we are discussing about that in the UAE, how we can grow apple, and it's an even harsher environment. And one key part for the apple industry, I believe, it's a bit colder than the UAE when I experience the weather, but it's to break the vernalization phase, which is needed in the apple tree, and also to choose the right rootstock. So I have not a very good knowledge of how farmers are using scion and rootstock in Kenya. It would be very interesting actually to look at that. And then based on our essay and also with my colleagues in Angers, it could be interesting to choose the right rootstock and the right cultivar. So there are cultivars that are resilient to very harsh environment, but at the same time, I don't know about the pathogen you can have and face, because in Europe we have a lot of Venturia. You have also the the different strain in North America. So actually, I would be more than happy to discuss about that. And we are discussing that in the UAE to try to grow apples there. So more than happy to meet you to further discuss this aspect. And also this question on epigenetic variations. We believe that some cultivar can be more resilient thanks to this methylation. So our aim with the study we are doing with my colleague Carol and Anthony, it's to see some epigenetic markers that could help us to choose some cultivars that are more resilient. And this definition of resilience, we have a lot of discussion about that, how to assess it, which cultivar could be more resilient beyond this vernalization phase. So I would be more than happy to discuss that further. Thank you. Thank you so much.
Yeah, so thank you. You can still log in and listen to the other presentations coming. So thank you very much. We'll have to listen to one of the presenters who is logged in also. So we will not have Tibrak as I'd promised. Let's just have one more presentation because there might be technical issues. It's good we get him to present. I don't know if Anthony, you're there? Is it Anthony? Professor. Do you have Anthony? Yeah. Dr. Baraza stepped out. It's not Anthony? Yeah. You mentioned of Anthony. Is it Mabele? Can he, let's see. Mabele, are you there? No? I think let's break. Dr. Baraza stepped out, so I'm not sure who is Anthony. Yeah, let's break and then come back. So we'll have a break for 20 minutes and then we continue. The theme will still be the same, genomics in crop improvement and production. Yeah, let's break, and thank you. the next session. The next presenter is Anthony Mabele. He will be presenting virtually, and the title is Fast Detection and Genomic Characterization of Fussy Bean Mild Yellows Virus Infecting Groundnuts in Kenya. Anthony, can you hear?
Yes, I can hear you. Thank you.
Welcome. You'll have 15 minutes and maybe 5 minutes for questions. Welcome.
Thank you so much. I need to share my screen. Uh, Can I go to full screen so that I can share my screen? Thank you. So, just a minute. I am there. Yeah, I hope you can see my screen now. Thank you. Thank you for, sorry for the delay. One, I'm Mabel Anthony. This is part of the work that we did with Professor Mieho from Masinde Muliro University of Science and Technology, whereby in line with the, yes, please. Yes, please. Oh, correct, correct. I can. Thank you so much. Hope that's okay now. Thank you so much. In my introduction, I was saying that in line with the theme of the session on the role of evolutionary genomics in addressing challenges in agriculture and environment in Africa, we have been doing also part of this research in legumes, and this was in groundnuts, whereby the title of this session for the presentation is about detecting Fussy bean mild yellow virus and characterizing it genomically. At the first report occurring in groundnuts in Bambara nut in Kenya. As I had mentioned earlier, with Professor Mieho, you were working on this, and we're still working on it. So briefly on the presentation outline, just in line with the normal routine, give introduction about the virus, then some of the statement, then justification, briefly objectives, then the hypothesis. But the most important, the material methods on how this research was done, present a few results, and discussion, then conclude and do some recommendation. You find that groundnut or peanut is one of the important legumes alongside Bambara groundnut because they are grown annually as food legumes in Africa and in Kenya. Basically, they boost on the nutrition ability and act as security earners for the farmers in terms of income. Ecologically, they grow in diverse environments between these subtropical regions. But one of the challenges find that their yields are not commensurate with the demand present in the field. And first bean malaria virus is one of the viruses that is causing these yield losses. And this is one of the polioviruses that's contributing to yield losses. So briefly about polioviruses, what are they? We find that they are responsible for emergence of new disease phenomenon across the world. Some of the examples of these poliovirus that have emerged, we have the chickpea chlorotic stand virus, the CPCSV. We have the cowpea poliovirus 1 and 2 due to coevolution. Then we have the cotton liver rot virus causing the blue disease. And also, I've also worked on cucurbit aphid-borne yellow virus, originally cucurbits, but occurring also in groundnut. And then our first been myelinovirus, the virus of discussion at the moment. You find that poliovirus have a narrowly relative host range, as reported by King et al. But Anderson, in his research, he mentioned that out of all the combination of pathogen that causes disease losses, viruses account for 47%. And we agree that once viral infection has occurred, most of them are irreversible. They may remain forever unless some plant health management technologies are put in place. Poliovirus are transmitted by vectors like aphids, and in this case, PBMYV, that is first bean myelinovirus, is transmitted by the groundnut aphid or the cowpea aphid, Apis crassifera majorly. And also, when you have grafted scion, they also transmit as done by Shaman et al. In Australia. I'm mentioning specifically groundnut aphid because you find that other aphid like Mysis pascae also, to a less extent, when we discover in the results. So there was no report for first bean myelinovirus transmission mechanically or by seed. And we did some screening on this, as you're going to check. In Israel, the paper-white fly-borne yellow virus is transmitted by a white fly, Bemisha tabeshi, is also a poliovirus. So you find that transmission of poliovirus also have wider vectors and also mechanical transmission. So just briefly on the problem, you realize that there's little understanding of this virus that we detected, or I detected, and you find that it had not been identified in Kenya, and it means there are limited management approaches that could be harnessed to increase the yield of the crop by reducing the pathogen. So in Kenya, where groundnuts are mostly grown to represent the country, and you find that poliovirus have potential permutations across the different agroecological zones because their virulence differs depending on the region. So, and that is going to be very important and has been going forward on genomic analysis to inform better plant health management technologies. So it's all about determining the distribution and molecular characterization of first bean in Kenya, and then map the incidence within the country and characterize to generate more knowledge. So we may agree or fail to agree, or reject or fail to reject the null hypothesis. So basically during field survey, these are photos of how I was conducting the research sample collection. Remember, it was immediately after COVID-19. That's why you see me in a mask. During the survey, which was systematic and purposive, because you collect symptomatic leaf samples and then store them in a cool box at 4 degrees until we analyze them in the lab while mapping the GPS. So these are map of the region that I studied. Then also this was the screenhouse trying to identify the indicator plants, which could be alternative hosts for the virus. And basically, I carried out research on 6 popular legumes, which are grown by farmers. At times, they intercrop them with groundnut, and it may form part of the alternative host. So, and then these were the insect-proof cages trying to establish vector transmission by other aphid like Apis coccibai and Mysis pascae, if they can also be including on the line of the biotypes which help in transmission of PPMYV alongside the groundnut aphid, Apis crassifera. And this was done after we had also prepared the inoculum in the lab. And then the data was analyzed, the field one by R software. Sequencing I did by next generation sequencing technology, Illumina platform. And then analyzed the sequences using MegaX software. So this is the sequencing platform. After the sample is collected, then RNA is extracted. I use the RNA-seq minicad Chiagen by following the manufacture protocol, quantify the RNA, then synthesize the DNA, then prepare the library on the platform, and then sequencing, and then I got my sequences. I did the quality check, and then through the novel assembly, blasted, then MegaX. I got the phylogenetic tree, and then try to. So in the field, these were some of the diverse symptoms that were present. You find that when we talk of mild yellowing symptoms, because originally this virus was occurring in Australia, in Mangbin, but then in Kenya, I detected in groundnut. That's why the title is fast detection. So some of the symptoms to a pathologist, these symptoms are very good, but to a farmer, this is disaster in waiting. And when you go to the field and you feel like you are like, wow, the farmer may wonder what's wrong with you. Are you celebrating because they are losing their crops or not? But anyway, that's part of research. And then see, when you uproot some of them, the yields, they are portless. You find on the left hand lower corner there. So this is part of the survey data analyzed. So this was part of socioeconomics. So this was ELISA trying to use Antisera to detect the presence before subjecting to PCR. PCR using specific primers for PBMIV, Facibin virus, and then 21 of the samples tested positive. And then these are some of the symptoms during mechanical inoculation that we saw earlier during screening. And then this one during caging. In the cage, you find that part A is for the groundnut, but then C is for the Bambara groundnut, which also took in mechanical inoculation of Facibin virus and then expressed some symptoms, which were mild as shown in D. And then B is for the healthy groundnut, but you find that the INS one, the apical young leaves were showing symptoms of Facibin. So during sequencing, you find that the Kenyan isolates clustered more closely with the Australian isolates, which were also detected from groundnuts or other legumes. And this one in genomics, we talk of identity by descent, which were more closely related. And it means there's need now for us to find out whether it's the identity by state or by descent. So this was part of the other Facibin. And Facibin, we use it also as a routing out group for the first phylogenetic tree. That's why I decided to include it here. So basically, briefly in conclusion, we find that Facibin myelin virus infects groundnuts, causing significant yield losses. And this is the first report of Facibin myelin virus in Kenya with a new host record that has never been reported in the country and also has not been reported in other legume like groundnuts, because in Australia, it's the only country where it was reported in Mangbin. So, and finally, I conclude that pests and diseases are still the main factor reducing groundnut productivity in Kenya as a country. So I recommend urgent need to establish the alternative hosts and the economic impact of this virus in Kenya, and also characterize it further so that we get more understanding on its persistence within the different pathosystems and severity at which plant stage, and also the vector diversity. So that is all I can mention for now. So part of the publication is on the first report, which was done in new disease reports by the BSPP. So we happen to be funded by the university, also British Society for Plant Pathology, Connected Virus Network, which is now at IITA, ILRI, KEFIS, and Zera Bartek Foundation, and also Plant Cell Atlas. Thank you so much for listening to my presentation. Thank you so much.
Yeah, thank you very much for the very interesting presentation. For me, it reminds me a bit on the early days of COVID, where you always assigned origin to those countries like South Africa, who had a lot of surveillance, and those which had no surveillance you notified. With that in mind, do you think this association of some of the viruses to Australia is really telling us something about infection history, or it's just because of the lack of other reference samples from Africa?
Thank you. Can I respond before you move to the next one? Yeah, thank you so much. At times when you go to the field to do research, you may detect unique symptoms in the field, and due to the advantage of advanced diagnostic platforms, you collect those samples, and because when you search in the database, when next generation sequencing detected first been malarial virus, the only place we could identify was in Australia. So the question is, because there are some viruses which are restricted regionally, like cassava brown streak virus restricted in the Eastern Central Africa. Groundnut rosette disease restricted only in Africa, despite groundnuts being grown globally. But then here is one virus which is being found in groundnuts, which is not its natural host. The natural host was detected in mung bean in Australia. So probably we could talk about either evolution of viruses or co-evolution of viruses and new encounter disease phenomenon, because if it's occurring in Australia and then it's occurring in Kenya, will we be sharing similar agroecological conditions? Could the vector biotas be thriving well? What could have enabled this disease to be brought in and had not been detected? So that's a question that we need to answer, and scientifically we can follow it up to unravel that. Thank you.
Any other question? Okay, one more.
Okay, so thank you, Anthony, for this nice presentation and nice work. So because this is the first case you are reporting on this virus, I don't know, what would you suggest some of the interventions? I know you've mentioned a bit of this, but critically what would be the main approach? Yeah, would surveillance be very critical in this, or especially in your sampling sites, because viruses are very unique in the way they emerge, and they are always very difficult to handle. Thank you.
Thank you so much for that wonderful question. At the center of all this is the farmer. We want to help the farmer not to lose the crop, and we also want to have abundance. And as we know, groundnuts being legumes, they always help in preventing malnutrition. And when our groundnuts are diseased, then we lose it. So the best intervention, since already this is a complete genome of first bean malarial virus, then at plant breeding level, we look at those virulent genes in the open reading frames and see which ones can we knock down, so that through advanced technology, so that we can enable breeding for resistant varieties, if not tolerant varieties. Because at the end of it all, just reporting it without any mitigation measures, as you put it, is just accumulating now more dust on the shelf of the knowledge that we always generate. And for plant clinics, to enable farmers know how to identify unique symptoms like viral symptoms, and then they can also report to the plant health officers so that they can mitigate and also look at the varieties. Because most of the varieties that farmers grow, they normally generate them, and we need to have certified seeds for the same also to mitigate over the same. Thank you so much. That's what I can comment at the moment. Thank you.
Thank you, Anthony, and thank you for your responses, your participation. Continue logging in and follow the presentations. One more?
Yes.
Okay.
Thank you, Anthony, for that wonderful presentation. Mine is very, very short. These incidents, I know farmers in the field grow different varieties of groundnuts. Was it on specific varieties or in all varieties of groundnuts? Because in addressing the intervention, would be looking at sources of resistance. What was the situation when you did the survey? Thank you.
Thank you. Thank you so much. That's a vital one. In the field, we have different varieties that farmers prefer to plant. The most common variety that was affected by first being mild yellow virus was red Valencia and the common red. But there was one variety called Manipita H1, the white-like. It appeared to be more tolerant to the virus severity because you find that the other ones were more severely affected, but Manipita H1 was more tolerant. And that brings us to the point that probably there are genes in some plants which can be tolerant, and if we can map them, they can become resistant. So if we can crossbreed and have varieties which are more tolerant and not more susceptible, like the red Valencia and the common red, as the farmers used to refer them, that can seriously help our farmers. There's a wide scope of knowledge over this. Thank you so much. You're welcome.
Looks like, yeah, those were the questions. Thank you again. Let's applaud him. Thank you. So, yeah. So our next presenter is here with us, Mr. Olivier Dayu from Germany is going to give a presentation titled From Gene to Field, Genomic Insights into Rami Kappa fistulosa for Managing Parasitic Weeds in African Agriculture. I think is the presentation with you? Yeah. So welcome, Olivier.
Okay. Hi everyone again. Yeah, my name is Olivier Dayo, and I'm originally from Benin Republic, that is in West Africa, and I'm pursuing my PhD degree in Germany at the University of Munster. So before I moved to Germany, I did my master's degree at Kenyatta University, where I was introduced into the field of parasitic plants by Professor Runo. And so today I'm going to talk about Rampicappa fistulosa, which is a parasitic weed that affects a lot of crops in sub-Saharan Africa. Yeah. So Rampicappa fistulosa is also known as rice vampire weed. And this parasite belongs to Orobanchaceae family. And that family harbors at least 50% of the world-known parasitic weed. And so this Rampicappa is known to be an issue in monocot and dicot. And studies suggested that yield losses inflicted by this parasite range between 23 to 70%. And economically, these amount to at least 82 million USD loss every year in rice industry alone. And that affects the livelihood of 140K household. And in this picture from left to right, you see these are rice fields heavily infested by the parasite. This is a close-up image of the parasite from a growth chamber. And this is soybean infected by the parasite. But how does the life cycle of the parasite look like? This parasite is really an opportunistic plant. First, it can live both as a normal plant or autotrophic plant, but it can also live as a parasite. So, like any normal plant, it can germinate, emerge above ground, and develop, set seeds, and then the cycle continues. But when the parasite senses the presence of a host plant, then it takes advantage of the host plant by attaching its hostorium. So this is the organ that is known as hostorium, which is a parasitic organ. It attaches to the host root, then hijack any nutrient, water, or sugar that's present in the host plant to its own benefits. And how does this parasite sense the host presence? It's by some hormones present in the host plant's root exudates, known as hostorium-inducing factors. And so the main problem here is that we don't have much knowledge about the biology of the parasite, and there was no genomic data to understand the parasite's biology. And the solutions here is first to try to grow the parasite in vitro and run bioassay or experiments, and also did some RNA-seq and proteomics stuff to understand the parasite better. And we also did genome sequencing, and all this combined together will help, hopefully help us to manage the parasites. And so what we did first was to grow the parasite on different host species. So here you can see palm millet, cowpea, rice, and tomato, which cover monocote and dicotes, and the parasite alone as control. And what we observe is that the parasite had a much better growth when it's with host, without compared to when the parasite is alone. So no host. And as you can see, well, the images are not of that good quality, but the leaves of the parasite in presence of the host are longer, and they are numerous compared to when the parasite is alone. And you see these individuals, they are vigorous compared to when the parasite is without host. And our estimation suggests that the parasite show at least a 3 better growth in the presence of host compared to the parasite alone. And second, we measure photosynthesis efficiency of the parasites on these different host species that I've just shown. And what we observe is that on some hosts, and that's palm millet and cowpea, the parasite had a higher photosynthesis efficiency compared to the low photosynthesis efficiency in some other hosts like rice and tomato. And what is more interesting here also is that the high photosynthesis efficiency in these two hosts is more or less similar to that of the parasite's photosynthesis when it has no host. And that suggests that the parasite can still maintain its photosynthesis ability depending on the host condition. We also score the number of hostorium attachments. So you remember I show a picture where you have the parasite hostorium attaching host root. So we counted those attachments, and what we observe is that on hosts where we have high photosynthesis efficiency, the number of hostorium attachment is low. But on hosts where the photosynthesis is low, the number of attachments is high. And what is also interesting here is that our analysis show a negative correlation between Fusarium count and photosynthesis. Of course, we must be cautious here because correlation doesn't always mean causation, right? And so what this suggests is that the parasites invest less in photosynthesis, so here, when it can form so many attachments, because the attachment help the parasites to get nutrients. Second, we study the cell-to-cell connection between hosts and parasites using microscopy and micro CT scanning. And what we observe across the different host species is that the parasite form xylem-to-xylem connection with each host. What does that mean? We know that in plants, xylem tissues help plant to get water and dissolve nutrients. And so the parasites just connect its xylem tissue to that of the host. It's like connecting two pipes. So anything that is passing through the host, the parasite is hijacking it for its own growth. Yeah, so this show a xylem-to-xylem connection conforming a vascular integration between host and parasite here. And second, we were interested in understanding or knowing genes that are regulated in each host species. And we collected the tissues of the contact zone between host and parasite. And then we sequenced the RNA, filter any transcript that belong to host, and then we perform a weighted gene co-network analysis, which clusters genes based on their expression pattern into modules. You can also call them as clusters. So these analyses show us 45 modules, so these different colors here, 45 modules, and also some of these modules had a correlation with either of these phenotype, photosynthesis and attachment. And what is really important here to mention is that we detected some hab genes. So those are the genes that are driving the expression, gene expression pattern in each modules. And these hab genes were upregulated in the host, where the parasite had low photosynthesis efficiency and high number of attachments, and those are tomato and rice. So among these candidate genes, we have GALS1, which is really important because before the parasite can invade the host tissue, it needs to do cell wall modification. And this enzyme here help the parasite to do that. The second thing is these last two genes, which is associated with oxygen transport and signaling. We know that oxygen is really, really important in the development of xylem tissues. And so by developing these, the parasite, as I showed you before, this gene helped the parasite to develop this xylem bridge to connect to that of the host to get nutrients. And another gene that a candidate gene that we found is this here, 55, which normally was reported in Arabidopsis to be involved in defense mechanism. But in the parasite, we believe, and that's our interpretation, that this gene is also helping the parasite to fight back the host in case there is a defense from the host side. So this suggests that the parasites can adapt its gene expression depending on the host species. And next, we also characterize the genome of the parasites by genome sequencing. And here I'm not showing the genome survey, but our interest here was the genome evolution. So the first thing we did was mapping the genome of the parasite against itself through self-to-self centenary analysis. What this helped us to detect is the genome duplication event. So here, on the x-axis, we have quantics from the parasite genome, and the same thing here. And so what these are showing are the events of whole genome duplication event in the parasite, and we have detected at least 2. And we complemented this with silent mutation analysis. And these pics here shows the different genome duplication events. And there, that is a noise. And so what this suggests is that in the genome of the Ramphicapa fistulosa, there is a genome duplication that is going on there, and this also expands what have been found in the parasite from this Orobanchaceae family, because in other parasites like Striga and Philibanchi ramosa, those are other parasites. It was also shown that those parasites undergo, their genome undergo whole genome duplication event. And yeah, we also study the function of gene families that were expanded, and we observed that most of these expanded gene families were associated with virulence factors, which help the parasite to invade the host. And auxin also, as I said before, it's really important in xylem tissue development. And another thing is those genes that are from that were gene families that were contracted, most of them were associated with photosynthesis. This suggests that the parasite is shifting from photosynthesis process towards a really challenging parasitic lifestyle. In the family of Orobanchaceae, it has been shown that before a plant becomes a parasite, it has to undergo 3 different phases. And the first one is genetic innovation, because a normal plant will not just develop Hostorium. And so where are the genes coming from, the genes that are used in the development of Hostorium? And so you are interested in the origin of those genes. And what we did is to map RNA-seq data of Arabidopsis, different tissues, to Ramphicapa genome, because we don't have RNA-seq data for all the tissues for Ramphicapa. And so what we observe here is that most of the genes were recruited from flower or root or seedlings. And that means the genetic innovation in these parasites is that it's obtain or co-opted genes from these tissues, duplicate them, and then remodel and use them in the formation and development of Hostorium. And so in summary, this study shows that Ramphicapa can modulate its physiology, anatomy, and genetics in a host-dependent manner. The second is that I'll also mention self-parasitism, which means that the parasite can release its own Hostorium-inducing factor. Normally, it should come from the host site, but the parasite can also parasitize itself. And that also means that the parasite lacks self-recognition mechanism. And this is a huge issue. That means it can attach to anything. And also this study showed that the integration of genomics and transcriptomics can reveal host-dependent reprogramming of the parasite's genetics. And our future plan is to knock down some of the candidate gene using host-induced gene silencing, and also to conduct pangenomics and population genomics to understand genetic diversity and the selection pressures that were present in this parasite. So I would like to thank my supervisor, Professor Susan Vike, my lab and the university, all the sponsors, and of course, the organizers of this amazing symposium, and in particular to Professor James for all the communication. Thank you very much for your attention.
Thank you, Olivier. And you see the weed you said is called vampire weed. So it's a problem, and thank you for your insights. So we'll welcome again, maybe 2 questions. We are also running late. Yeah.
you said there are duplicated genes that have now, so the duplicated set, they get a new function in the Hostorium development.
Yeah.
So that means there are, like, in a simple example, there are two genes, the ancient gene and the new gene. So can you compare them? Because you should then also see probably signatures of selection in the duplicated one because of the new functionalization.
Yeah, that's a really good question. Though we haven't compared all these, but one thing that we also want to do is first to go and do this positive selection analysis to see those genes that were under positive selection pressures, and then see what we can conclude from there. Yeah. Thank you.
Yeah, and then Olivia, if I can ask, did you had in your title genomics? Did you do any or it's still going on?
Yeah, actually, I have not presented data here. I'm running another project where I'm comparing the proteome profile of different parasites, and that is completely another project that the paper is already, but I've not presented it here. Sorry.
Thank you.
Yeah.
Maybe one? Okay, if not, thank you very much. Let's applaud him. Thank you. So our next presentation will be virtual. I hope he's logged in. Is he from Nigeria? Salkalaini. I had seen him initially. If not, let's welcome Nahaman. Are you here? Yes, welcome. So Nahaman Arodi will be giving a talk entitled Evolutionary Genomics of Structural Disorder Reveals Mechanisms of Virulence in the Rice Blast Pathogen Macnapoteorizia. You'll come and pronounce it well when he gives the presentation. Let's have his presentation, Lordy. It's there. Okay, welcome, Nahaman.
Thank you very much. My name is Naaman, PhD candidate at the University of Eldoret, and I'm working with my team, Professor Miriam Kinyua, Amy Chepkwach, Paul Njiru, and Wilson Oyange. Paul Njiru and Wilson are based at Mwea. Welcome. I'm working on rice, and the disease I'm tackling is Macnapoteorizia. It's a very big problem. Rice is a very important cereal, second only to wheat in the world, and about 60% of the world population is dependent on rice. In East Africa, it's around 45 to 80,000 tons are produced annually, and this is a symptom of rice infested with blast. It attacks many parts of the rice, and thereby interfering with the yield, the leaf, the collar, the neck, and the panicles. It manifests itself in so many other forms, just like our presenter talked about the vampire. Here is another vampire for rice. So, because rice is food for majority of people in the world, you can see how it is spread all over the world. North America, South America, Africa, where rice is grown, presented in green, Europe, Russia, and even Australia. So it's a worldwide problem. The reason being, breeders are usually attempting to do resistance breeding, but the resistance breeding is always being broken down because of the evolutionary arms race, the survival for the fittest. The fungi keeps on attacking rice, and resistance in resistant lines or improved lines are often brought down. But I'll narrow in later on into this particular antagonistic kind of behavior with respect to basmati rice. Basmati rice, which is also popularly known as pishori, ranks number 1 because of its good grain quality characteristics, but it is defenseless. So it is you hit, I hit back. Look at the cartoon. I hit back. And the winner harvests all, the one who is very strong. And it's because of evolutionary arms race, as we're going to see. So this study has been founded under the gap where comparative disorder profiling between fungi effectors and rice immune receptors remains limited. During host pathogen attack, it has been reported that the pathogen releases a set of batteries to attack the host plant. In return, as you have seen earlier, the plant also has defense mechanisms to antagonize it. And these proteins, known as effector proteins, are able to evade plant's immunity because they are 2D structures, they are open structures. The plant immunity is not able to recognize them. So we refer to them as IDPs, intrinsically disordered proteins, because the plant is not able to recognize them and therefore cause immunity. So the hypothesis is that the virulence associated proteins in Magnapothe rice are evolutionarily enriched in intrinsic structural disorders relative to host receptors. They're highly disordered to the extent that the plants are not able to recognize them and therefore have a free will during the inversion. Our objectives, one was to compare disorder properties of fungal effectors and rice immune receptors, to determine whether structural symmetry exists at the host pathogen interface, and explore evolutionary implications of intrinsic disorder enrichment. As I've said, basmati is very vulnerable because it has no soldiers to defend itself, and therefore the Magnapothe orisa has a free will. You see, another problem is it has been very difficult to form interspecific crosses using basmati because it has jealous genes. Often, the F1 hybrids are sterile, and therefore breeders are not able to develop resistant inbred lines for use in basmati rice improvement. The dogma is, why is it defenseless in this arms race? And therefore went ahead to analyze for the category of effector proteins versus the receptor proteins during this particular interaction. This is what happens for a resistant line. Resistant genotypes, when faced with the blast fungi, they initiate what is called effector-triggered immunity, and therefore they remain resistant. For further studies, these particular phytohormones get upregulated. These particular phytohormones get upregulated in resistant varieties. But as a staple variety like basmati, effector-triggered susceptibility is observed. Auxins as a phytohormone is accelerated. Auxins is upregulated. That can be a very good candidate for next studies and their associated pathways. How does Magnapothe effectors evade rice receptors? This is just illustration of what happens. Through recognition, effector is recognized by R protein, then evades the cell and triggers binding, hence causing immunity. Two, it can also evade using the ID proteins, protein-protein interactions by gene flow analysis. If the highly disordered means no recognition and free will, and also they can do suppression, where they suppresses the plant immunity, thereby causing susceptibility. So it is ever in the arms race. IDPs shape shifters of infection, causing flexible, elusive, and are very powerful. Our approach, first of all, we used dry lab bioinformatics approach. Many of these crops have been sequenced, including rice. Rice is so much over sequenced, and so is the Magnapothe oryzae, which is causing the disease. The data is there. What is to do with the data is a problem, and the data is too big and too large. So we extracted the data using bioinformatic approach from NCBI, carried out effector gene identification, SNP analysis, and looked at the protein disorder profiling using various bioinformatic tools and pipelines. This is how the pipeline looks like. Begin with, we begin with the identification of effector proteins. We use the genome analyzer to predict segregated effector candidates using either of these tools, or use NGLA. Then effector filtering. After filtering, we decide how disordered they are. The disorder pattern was done through a threshold of 0.5. Any protein with a threshold of 0.5, above 0.5 was considered disordered, and the ones below 0.5 are ordered. It is disorder that causes virulence. Then we went further to look at structural characterization, then functional annotation, conservation analysis, and then integration and protection using these all particular tools. We used effector P to identify the effector proteins. We used IU predictor 2A to determine the signal peptides and which regions were disordered. We used AlphaFold 2 to look at their structural analysis to either 3D or 2D, and then we analyzed the whole data using Python and the Conda terminal in Python. What were the results? When you look at this, we found that the number of disordered proteins in the fungi were higher in number. Look at even the mean in the box plot, in the violin plot rather, as compared to the base of the rice genome. So there are more disordered proteins in the fungi and less in the rice, and that is why the fungi probably is having the free will and is ahead in the fungal race. So fungi proteins show a broader and higher distribution of intrinsic disorder. Rice proteins are mainly considered at the lower disorder levels. This means that higher disorder in fungal proteins suggests greater structural flexibility. And this flexibility may help fungal effectors adapt, interact with multiple host targets, and evade immunity. Rice proteins are more ordered, reflecting stable immune recognition and signaling. So the fungal effectors are more intrinsically disordered than rice proteins, and the structural asymmetry exists between the flexible and host stable proteins. This was further explained by the disorder score. When we compare the disorder score and the frequency length, we find that proteins above this threshold, this is 0.5, above here are disordered, and below this line are ordered. And it's a disorder that causes disease. So many of the disordered proteins were between 0 and 2000 length in terms of amino acid. However, a few are found beyond 300 to 400 here. We realize that these ones are known motifs, known genes that are being used usually by breeders for breeding for tolerant breeding purposes, and mainly they are tripeptides. The ones here are mainly dipeptide, short sequence amino acids. This is responsible for what we had seen earlier. Again, we cannot talk about the asymmetry without mentioning the tripeptides, the dipeptides. The bipeptides are the short protein sequences which are known to be folded very easily to cause virulence. So, among the proteins, we only selected 20. There are so many dipeptides. There are battery proteins produced by almost 546 genes. So the first 20 are of interest, and the most common one is the alanine. We have used the codes because the two protein dipeptide, A plus A is equals to 2 protein sequences. They're the ones which are easily used by the pathogen to evade plant immunity. So the peptides are center of great interest for this particular research. Again, when you consider their frequency and diversity, again, many of the IDPs were found above that particular level. I mean, they are less diverse. They are less diverse. So great diversity was associated with lower mean frequencies. Lower diversity was associated with mean diversities, and there's a weak correlation between diversity index and mean frequency. So effector dipeptide diversity increases as average frequency increases, suggesting a trade-off between repertoire breadth and the dominance of frequently observed dipeptides. Again, not very important, but we have to look at the tripeptides. Many of them are known. Again, there is no clear relationship between tripeptide mean frequency and diversity because they are not off, they are stable. They have a 3D structure and therefore they are not easily evolved and used to cause virulence. But as compared to the dipeptides rather. Again, the most frequent tripeptide was the same alanine, and therefore it is the most protein to be studied towards tolerance or resistant breeding. Our key take-home message is that virulence proteins exhibit elevated intrinsic disorder, and disorder likely facilitates evolutionary adaptability. Structural asymmetry reflects host pathogen coevolution, and intrinsic disorder is an evolutionary feature of pathogen virulence. I want to thank my mentors. Some of them are seated with me here. The Max Planck Institute and the Nicole Lovino Lab, where I'm doing the bioinformatics analysis. Thank you very much for your attention.
So thank you, Naaman, for your presentation. Again, we'll welcome maybe 2 questions. Any question, comment? Okay, any question?
Thank you, Naaman, for that beautiful presentation. You've taken the direction of the dry lab, considering the fact that rice has already been sequenced and a lot of sequence information is available. So this is something that with the sequence information, knowledge on bioinformatics would be a beautiful thing to be able to actually establish what those sequences tell us. So you've already highlighted to us how actually this basmati rice is so susceptible to the disease. Does that declare doom to this favorite food for the Kenyans and the world? Because anybody going to the market, going to the restaurant, gets basmati, is very happy to have it. But looking at it from the perspective you just presented, is there any hope on this particular basmati? Thank you.
Thank you very much, my bioinformatician lecturer. Actually, Prof is the one who introduced me to this concept, which has taken me places. There is hope. There is hope, and in my second objective, my field work, that is also I'm doing, which is not represented here. I subjected the fungi, the whole fungi genome around 3,125, and compared with one of the resistant lines found in Philippines called Nippon berry. And we realized, I've already seen that through bioinformatics analysis, genes which are being upregulated. The Nippon berry is tolerant to all the strains of the fungi, but it doesn't have good grain characteristics. It has got low yield, it has no aroma, and we want to ensure that the basmati that we are going to improve has all the characteristics that are desirable in the original basmati. So I've designed already a back-cross breeding program where I'm going to introduce Nippon berry 3 times. Once. Nippon berry becomes a donor, then basmati is integrated 3 times the system to recover as many as 98% of the basmati characteristics, so that I can have lines to be used in basmati rice improvement. Nippon berry has been studied to be tolerant to all the viral resins, and I've identified 4 proteins which are upregulated. We interrogate the genome of Nippon berry together with the fungal genome, and has given us points into leads at why there are signal peptides at that particular locations. Thank you. So there's a very big hope. Similar studies have been done elsewhere in Japan and China, but in Africa, none. Another approach has been on gene editing. I think China and India have released a gene-edited rice, basmati, which is resistant to this particular problem. Again, there is something called Pusa basmati in Asia. Pusa basmati 1, 2, which has been bred using basmati as a parent together with other tolerant varieties, and already tolerant to the blast disease. However, the resistance has always been broken 2 to 3 years. Thank you very much. Thank you very much.
Thank you. It's okay. You had something to say?
Yeah, because of the co-evolution, but not with the Nippon berry. Thank you.
Thank you. Any other question? Yes.
Thanks for your presentation. I think it's always good to have hope. But parasites are usually also evolving quite fast. They have a short generation time. They have a huge population size. So that means anything that you might do on the side of the host, parasites might be able to adapt quickly to that new situation. What is your take on this?
That formed the basis of this particular study. We're actually looking at why is the this pathogen more fitter. From previous studies on resistant breeding, the resistance has been broken in 2-3 years cycle, and we are trying to unearth why is this resistant breeding towards blast is not durable. It is because of coevolution. The pathogen is too fitter, and the fitness is because it has got more of the dipeptides, the short sequence amino acids, which are highly disordered, that the plant host, today, you attack me this way, with this arsenal, I release another arsenal. The pathogen release a dangerous weapon in Iraq, US war now. You think you have cleared the enemy might just even stronger. Again, there is that aspect of the work of what is called conditional genes, epigenetics. This particular fungi sometimes is more vulnerable in certain ecologies than in others. And we don't know why in zone A, it is more severe, and the same crop, and in zone A is less virulent. And epigenetics is part of my studies here, part of my objectives, which I'm going to study when I expose my treatments into different agroecologies and subject them into pathogenesis, both in the greenhouse and out of the greenhouse. Thank you. Of course, if you're going to have a durable resistant, we shall have no work. And then this theory is also backed by what is called survival for the fittest. If you look at even the chromosome number of the pathogen is so less compared to the chromosome number 12 of the rice. So it's about survival, which is part of the theme of coevolution. Yeah, thank you very much. Thank you very much.
Yeah, thank you, Naaman. And as we finish, are you the one who did the illustrations there? They are nice.
Nowadays,
it's well illustrated.
Yeah. I was assisted.
Oh, okay.
To some extent, I used AI. Get this one, so it's allowed, though I did not cite anyway. I'm supposed to. Yes,
thank you.
Used AI to at least generate my concept and idea. Yeah, to help me bring a better work. Thank you.
Thank you. Thank you. Let's applaud him. Thank you. Thank you very much. Thank you. So we've come to the end of the session. That was session 3. We are going to switch to session 4. And I'd like to invite the chair, Dr. Kennedy Kania, to come and continue. It's been a pleasure. I'm a medical biochemist, so I was learning some stuff, but we appreciate. Genomics, proteomics is cutting across. The approach is really the same in all aspects. Thank you.
Thank you, Dr. for that particular session. I think it has been a very interesting session for us. And I want to appreciate this symposium, the manner in which the organization has been done, where things are moving systematically. We have been in the terrestrial kind of research, crop improvement or whatever. So we want to transition to another very exciting area of research, and this is in the area of aquatic environs. This is to look at what in Kenya we famously call the blue economy. So like we've been looking at the terrestrial economy, we now transition to the blue economy to see how this integrates together in our symposium to be able to actually focus the themes that have been laid for this particular symposium. So our keynote speaker for this particular session is with us, Professor Cyprian Gatongo. Professor Gatongo is from University of Zambia and is a renowned researcher, a renowned academician who has background in biology and chemistry I know we were having a small chat last evening over tea. He mentioned how he was in the breeding area, and we had very interesting discussion there. And then he has again moved to another interesting area. This is the aquatic environment, where he is now working with fish and whatever. So Professor Gatongo has an MSc in applied genetics from Birmingham University, UK, and a PhD in the University of Graz in Austria, based in evolutionary biology. That tells you how widely research he has done. We know he's a Professor Gatongo is also a vice president of the Institute of Applied Science in Zambia and in other various disciplines, which tells us that we have the privilege to actually have one of the best in this particular area to be able to guide us. So he's going to take us through the title of the talk, known as Unlocking the Evolutionary History of Zambian Fishes Using Cyclic as a Model. So there would be no other beautiful way to be able to introduce this aquatic environment than to have Professor Gatongo. Karibu sana, Professor.
Thank you very much, Chair, for that introduction. And good afternoon, colleagues. I know we are approaching lunch, but we can still make it between now and lunch. Yeah, as the Chair has introduced me, I have actually worked with plants, and I was doing things related to plant breeding and conservation, but also there we are using molecular techniques, but based on plant genes, especially the chloroplast genes. So I transitioned to fish genetics for my PhD, and I've stayed with fish. But sometimes I also work with plant colleagues. So those are my contact details in case they disappear from the screen. But I'm sure these things will be shared. These presentations will be shared. That number is a WhatsApp number, but you need to put the Zambian code. But the email is just as presented. So I'd like to start by acknowledging collaborators, including University of Eldoret, who are the hosts of this symposium, and the SMBE, who are the funders, and my previous experiences at different institutions, including universities, and I also acknowledge the support I get from my government. So if you see the left bottom corner. So the layout of the presentation is as outlined. I'll try to be general, talk about East African Great Lakes and the cichlids based on literature, but also interactions with the colleagues from Switzerland, Austria, Germany, Belgium, and South Africa. who have explained some details of the morphological adaptations shaping cichlid speciation. They have also talked about environmental drivers of this radiation and also genetic drivers. But my own work starts with the evolutionary history of Zambian fish, where I actually did the work myself. And I also looked at hybridization between native and non-native Oreochromis species. So this Oreochromis is a tilapia. I think it's a farmed fish, but it's not native to Zambia. But to Kenya, I think it's native. So you don't have that discussion about the Oreochromis, especially Niloticus, like we have in Zambia. I'll try to talk about challenges and way forward, but mainly way forward, because challenges are well documented. So the Great Lakes of Eastern Africa and their cichlid fishes, I'll focus on the three lakes, Tanganyika, Malawi, and Victoria, and I've put in the relative ages, Tanganyika being the oldest, Victoria being the youngest. And in terms of the depth of the water bodies, Tanganyika again is the deepest, and Victoria is the shallowest. And these, all these lakes were formed by tectonic processes as part of the East African Rift Valley. I think we are either in the Rift Valley or on top of the valley, or at the bottom. I'm sure we are on top. So near here should be the valley itself, but we are on the plateau side or arm of the Rift Valley. So the valley is formed by ripping sections of the continent apart and creating deep basins which are filled with water. And they also disrupt the flow of original rivers. So rivers were flowing, then suddenly you have a Rift Valley, so the flow is discontinued. Either the river changes course or it disappears. So these are just diagrams of the Rift Valley. So there are two arms. So within the right arm, you have Lake Turkana and Lake Malawi. The left arm, Lake Albert, Edward Kivu and Tanganyika, and others. Lake Victoria is not really on the spot of the Rift Valley. It's in between the two depressions. I don't know where we are here in this room, but I think we are on the right side, and maybe between, let me not speculate, but we are somewhere within there. So that's another version of the Rift Valley. So if you look at the numbers of fishes, Tanganyika, 250 cichlid fishes, Malawi, 800, Victoria, 500. These are really large numbers of one, let's say, family. And these fishes have evolved into many species because of their different, or rather because their different phenotypes are adapted to different environments in rivers, lakes, and wetlands. And the phenotypic adaptations among these cichlids are expressed through body shape. These are morphological characteristics, diet, choice of behavioral, color, and behavior. So color, I think, or determines, say, sexual selection, because females are able to use colors to select their potential mates. And the phenotypic variations are due to heritable changes that take place in the genetic makeup. So the genes actually change, and then they induce phenotypic changes. And the changes at genetic level would be through recombination, mutations, and other changes. Someone mentioned the transposable elements. These also affect genetic changes. And once these changes happen, then they are subject to natural selection in the environment. So those changes which are best adapted survive. Those which are not become extinct, or their fitness is low, so they don't reproduce. So due to their rapid speciation, cichlids have been used as a scientific model in the study of evolution and speciation. So the speciation of these fishes has been very rapid in just a short period. So here I demonstrate some morphological adaptations of these fishes from the 3 legs. Yes, I want to ask how to create the red light on this. Top one, okay. Yes. So the Victoria, Tanganyika, Malawi. This is from that publication. You see that there are a lot of colors, a lot of shapes in these fishes, and there's some kind of parallel speciation or parallel evolution, where you see some fishes with certain types of lips, thick lips, appearing across, and you see elongated forms appearing also across. So although these are in different legs, but they seem to be evolving in parallel. So among these, you also see some riverine fishes. I think this is Orochromis niloticus, which is endemic here, and this is Astastotelapia burtoni, which is a riverine fish common around all the lakes. So among the ecotypes, you can see there pelagic or zooplanktivores, pedophage, eating young ones, snail crushers, lobe-lipped, which are insect eaters. Strange, I can't see the thick lobed-lipped ones easily from this angle, but they are there, and the riverine ancestral types. So it is assumed that most of these fishes which invaded the lakes originated from rivers. So when the Rift Valley formed and the lakes came in, so the river forms seeded the new lacustrine environments, and there the speciation took place, because in rivers, the environment is general, so you find generalists. But in the lakes, you find different ecological opportunities: rocky, sandy, deep water, muddy bottoms. So all these opportunities create the drives. So this paper talks about adaptive radiation of cichlid fishes in Lake Tanganyika, a morphological perspective. So here you see trophic morphology, like decoupled oral jaws and pharyngeal jaws. So in humans, we have just one set of teeth, the upper jaw and the lower jaw. But in these cichlids, you have in the throat another jaw, the 2 jaws there, which also help to crush, let's say, snails. So in terms of gene duplication, etc., you'd agree that the pharyngeal jaws would have come from genes which were for just ordinary bones, neck bones, but due to gene duplication, the new genes acquired a function to develop jaws in the neck. Body shape, variation, sexual dimorphism, mouth brooding, swimming modes, etc., are all variations which differentiate the fishes. Body size, you also have big individuals, small individuals, and here I include shell-dwelling. So shell-dwelling fishes are very small, so they are able to live in shells of snails. Not a big fish can live in a shell. So they are sheltered there, and they become their type. Male body coloration and color vision. So on the male body, especially on the anal fin, you have these egg spots, which are dummies. They are not eggs, but the female thinks they are eggs, and it tries to go there to lay her other eggs near those eggs, and in the process, the male fertilizes the eggs. So the color of the spots discriminates species. So certain females are attracted to certain colors. And also the diet choice separates the species. The nervous system also, the brain size, courtship behavior, brood care, social behavior. Based on the brain size or brain function also creates different species. Environmental drivers, like I've already talked about deep lacustrine environments, which means you have these deep lakes forming in the rift, filling with water and then creating different environments. Some are cliffs, others are rocky, etc. So different fishes adapt to different environments, and you have different species. Lake level fluctuations. There is geological evidence to show that there have been dramatic fluctuations in lake levels, especially low stands, caused by dry weather repeatedly. So when the lakes dried up and then filled, or when the level was so low, some fishes, let's say, split into different groups. So they speciated, and then on secondary contact, there were different species that couldn't reproduce anymore. And the empty niches created by these big aquatic environments also created opportunities for speciation. So you have also this habitat diversity, which I've already referred to. So just this paper also talks about lake level fluctuations. Title is that lake level fluctuations synchronize genetic divergences of cichlid fishes in African lakes. So if you look at the three lakes, when Victoria dried up, I think there was just a small water pool there. But when Lake Tanganyika dried up, you have three basins. For Malawi, you had that deep basin. So as the climate changed, these water bodies would dry up and then fill up. And this would be between 57,000 and 12,400 years ago. Lake Victoria dried up completely, and Lakes Malawi and Tanganyika were really low, and they just remained with those deep basins. So later on, about 11,000 years ago, the lakes were filled up again, and because of the separation during that dry period, they separated populations of fish, speciated, and when on secondary contact, they became different species, which couldn't reproduce the way they would have done before the drying up. So a multitude of species originated in lakes, Malawi and Victoria, with an impressive degree of morphological and ecological differentiation. So if you look at Malawi and Victoria, these are young compared to Tanganyika. So Tanganyika has very old lineages which have persisted, but for Malawi, most of the radiations are from one lineage. They are haplochromis. So in Malawi, I think they are called imbunas. In Kenya, I think, or in East Africa, I think they're just called haplochromis. So these are the ones you see in large numbers. But in Lake Tanganyika, you have 12 tribes, and one of them is haplochromis, or haplochromini, and it is speculated that the haplochromini seeded the two other lakes because they are younger. So, and the haplochromini maybe started off as a riverine. So because it could move into small streams, it found its way around the area, including the lakes. So patterns of genetic divergences of stenotopic organisms provide valuable feedback on geological and sedimentological time estimates for lake level changes. So here we collaborate the evidence from geology, sedimentology, and genetics, and behavior. And if you have fish or any other organisms which have a very narrow range of environment where they can survive, then they are very territorial, then you can infer some time estimates. Those were environmental. Now genetic, I think I've talked about this already, hybridization. So when two populations come together and are able to hybridize, then you have the F1, which has heterozygosity, and this heterozygosity is a start of variation. So once you go to F2 and other generations, including back crosses, then you create separate populations, some of which can become distinct species. I already also talked about transposable elements. These are moving genes. They can move from one position of the genome to another, and by movement, they might either disrupt gene function or activate a gene which was originally inactive. Maybe the regulator sequences would come near an inactive gene. I think I've talked about gene duplication, and 280 duplications were identified in some genes in cichlids, and as I've said, and already has been said, that you have the ancient genes and the new genes, and the ancient genes will continue being faithful and continue providing the normal function, and the new genes are able to try out new functions, and that's how you see new innovations. And one of the genes which has been studied in details is the opsin gene, to do with vision. And as I've said, vision determines maybe sexual selection and also diet and also navigation in the aquatic environment, especially in clean water lakes like Tanganyika. I don't know about Victoria. There's been a lot of eutrophication in Victoria, and I think the fish there cannot effectively use opsin genes to see, to navigate their way through eyes. Maybe they use other cues. But originally, all the lakes were clear enough. This paper is about ancient hybridization as it fuels rapid cichlid fish adaptive radiations. So here, just from the phylogeny, it has been inferred that we started with two almost riverine species which had contact around that hybridization zone around Lake Victoria. So you had the Upper Nile types and the Congolese. And when they hybridized, the hybrids were fertile and able to colonize the rest of the water bodies, the rest of the Nile and Congo and the Zambezi. So hybridization is one of the causes of new gene combinations, and it can lead to speciation. Transposable elements, I've talked about this, and there's this paper which talked about movement of transposable elements contributes to cyclic diversity. I think, as I've explained theoretically, that these transposable elements move around and they can either trigger the expression of the gene or disrupt. So because of this, you have new gene combinations, and these can also lead to speciation. And this paper was based, I think, on Lake Malawi, where they looked at transposable elements that correlate with opsin gene expression. So that was, let me say, the general overview of the speciation of fish of this group, the cichlids in Eastern Africa. But my own work, I think, starts from here. So I did work on Zambian fish. This will be the layout. I saw the gap, then did a phylogeny and speciation of the genus Pseudocrenilabras, then collaborated with the colleagues and did radiation of the Paleo Lake Makadikadi. Then I also did evolutionary history and biogeographic affinities of the Serranochromine fishes. These are also cichlids, and they are a member of the Haplochromini. So they are a sister to the Lake, not Tanganyika, here, Lake Victoria, and also Malawi, Lake Malawi Mbunas. And here they are called, I think, haplos. Then I also looked at rapid adaptive radiation in Lake Mweru, because we have two lakes on the Congo River, Mweru and Bangweulu. So we looked at both lakes and found that Mweru had a radiation, while the other lake nearby didn't have. So the gap I saw was that initially freshwater fish research in Southern Africa was mainly conducted on the Orange, Limpopo, and Zambezi basins, and ended at Zambezi River, and did not include Luapula Congo, which means studies were done there, and there, with the tributaries, but it did not include that system. So at the time I went in to do this work, you either had data for this part or data for that part, but not combined. But then Zambia sits at the watershed of two of Africa's major rivers, the Zambezi and the Luapula Congo. As you can see, this is a major river. This is the Zambezi flowing to the Indian Ocean, and Congo flowing to the Atlantic. It compares very well with the Limpopo and the Orange there. The Nile, and that's a blue Nile, that's a white Nile, originating from Lake Victoria, this one from Lake Tana. And here you have Niger River, Volta River, Senegal River. So these are the major rivers in Africa. So I was sitting just on the watershed of two of these major rivers. So we wanted to know in which directions fishes originated. Did they come from the Indian Ocean to the Zambezi and then seeded the Congo, or the other way? It was not known without combining the data. So, therefore, my research was an evolutionary study of a combination of cichlid fish from both the Luapula Congo and the Zambezi. So I combined the Congo and the Zambezi. So my sampling effort, this would be the Congo, drainage, the Zambezi. So if you saw on that major map, the Zambezi and the Congo are just shown as just simple lines, but these are river systems with the different other rivers and tributaries. So I sampled from the places labeled. So the first was a phylogeny of the Pseudocrenulabrus. This genus has a continent-wide geographic speciation according to river system. So all the way from Egypt to South Africa, you have this genus. But in each region, you only have maybe one species because they are generalists and the rivers don't have so many opportunities. For example, the multicolor has been reported to be confined to the Nile River. Like here in Kenya, you would find the multicolor. Nikosi in the Congo, but the philander is what you get in southern Africa, including my country, and other southern African countries, Namibia, Zimbabwe, South Africa, etc. So from this phylogeny, you can see, I don't know if it's clear, but we see that these are from the Zambezi. Oh, most of these are from the Zambezi. This is the philander. Then you have from the Chambeshi, which is the Congo. You have this species, and these are also from the Congo system. So in terms of origin, from this phylogeny, which has been rooted with the Nikosi and the multicolor, we see that the basal clade is from the Congo. And the upper clades are from the Zambezi. So we can infer that these fish started from the Congo and then invaded the Zambezi from this phylogeny. So multicolor, just to illustrate, would be confined there, and then the Nikosi somewhere there, and the philander would be in these river systems. But for the Congo, I think the lower Congo would have philander, but upper Congo would have, is it the other way? This is upper Congo, then lower Congo. Because of the rapids and so on, you can't navigate the Congo all the way from Zambia to the Atlantic. You find a lot of rapids, etc. Even the fish face the same barriers. So philander is not found in that area. So this is the paper which, out of a collaboration on the Paleo Lake Makadikadi. So from geological evidence and looking at the fish, we infer that there was a lake in that area, which is no longer there. Nowadays, we just find salt pans. And when you look at the fish, you see a surprisingly high number of cichlid fish species compared to other rivers. And the fish numbers in these rivers around Southern Africa compared to Lake Victoria. So suggesting that there must have been a radiation also in this area, most likely in a lake which is no longer there, and when the lake dried up, the fish escaped, took refuge into the surrounding rivers. Because the lake provided the opportunities which lacustrine environment normally provides. And when that lake dried up, the fish sought refuge in the surrounding rivers. So that key gives you the different river systems: Upper Congo, Middle Zambezi, Okavango, Kinene, Limpopo, Lower Zambezi, and Middle Kasai. Yeah, just to explain how that lake might have disappeared, because you see here, this is the Zambezi, as we know it. But according to geological records, this Zambezi, it was not the way it is now, but it was flowing into the Limpopo and then to the Indian Ocean. But now, it doesn't go all the way to the Limpopo. It changed course somewhere after the Victoria Falls. There was also some geological process which caused some depression. So the river changed course and joined there. And this area would be the site of the dried up lake. So once the river changed course, there was no more source of water, so the area became dry, and the fish escaped into the surrounding rivers. So this publication was on the selenochromines. These are related, as I said, to the haplochromines of the greatest African lakes. And there has been speculation that these are the probably ancestors which seeded these lakes, because they are haplos and they are found in rivers, so they move around, and maybe they are potential ancestors. And from this phylogeny, we also see that the tendency is to seed from the Congo to the Zambezi. So like here in this clade, you have Luapula Congo and Upper Zambezi, and so forth. But in some cases, you see Zambezi being ancestral to Congo, and the explanation is that when the Makadikadi dried up, which is in the Zambezi system, some of the elements seeded the Congo because it was in the vicinity. But generally, movement is from Congo to Zambezi. Mentioned that we compared two lakes, Mweru and Bangweulu. They are both in Zambia, and they are both along the Congo system. If you come on the Chambeshi, then Bangweulu and Rusiri, after the swamps, again the river builds up into the Congo, Luapula Congo, Mweru, and out into DRC. So these two lakes have the same environment, but we see more variation here. Yeah, if you see these arrows pointing to common things, but we see more species in the Lake Mweru than Bangweulu. And the explanation is that Mweru is a tectonic lake and is part of the Rift Valley system, whereas Bangweulu is a cratonic lake, which was either formed by impact crater, by a meteor, or it was just sinking and being eroded, but it's not a tectonic lake. So it doesn't have so many opportunities, and it's shallow. And these are the groups of fishes which I explained. And among these, we described some species new to science. Just an amplification. So we described one new species of Pseudocrenirabras from Lake Moilu in cluster 3. Let me correct, in cluster 5 actually, from this cluster. And then we described two new genera from the Lufubu, and these are the two individuals representing those two genera. So this is a comparison of the Philander, which was originally thought to be the only one, but we described this one related to Philander, but we call it Pyrocodalis. It's a different species, even on the phylogeny, even this morphology, and we followed the standard keys to describe it. We also described these two new genera, Paleoplex, new genus, Lufubu chromis, new genus, found in the Luongo and Kalunguish River and Lufubu River, respectively. So we also looked at hybridization. Like I said, in Zambia, Odochromis niloticus, or the tilapia, which is native to the Nile system, including Kenya, is used for aquaculture in Zambia, and it's hybridizing with the local Odochromis, and it's a problem. It's an ecological problem, it's a genetic problem. So we wanted to show if there's genetic evidence of this hybridization. So we took pure Andersoni, which is a local species, pure Macrocha, and we also looked at morphology. So these are the three natives, Andersoni, I think this is the Niloticus, should be Macrocha, and this is a hybrid. So a hybrid is supposed to have a combination of the traits of the two pure breeds. For example, the three spots, I don't know if it's imaginary here. And if you look at Niloticus, the tail fin has stripes, and you can see them here. So spots and stripes is a combination of Andersoni and Niloticus in this case. So here we see using PCA, the split between the different species, Macrocha, Andersoni. And then if you look at the hybrid and the Niloticus, you see a lot of similarities in the morphology. So it's very likely that the Niloticus is either already back crossing with the Andersoni, or that the shape or morphospace favors potential hybrid, further back crossing with Niloticus. So which means Andersoni is under threat just from this affinity here. Finally, I can talk about opportunities for evolutionary research in Africa. We have potential for continuing study of human evolutionary history. There's this theory of human origin. Here in Kenya, I've seen a t-shirt, someone saying humans originated from Kenya. In Ethiopia also, they say the same. So, but we can say maybe from Africa. So we are sitting, on a continent where everyone should come and learn. If you look at the diversity of the genome among humans, Homo sapiens, I think it's highest in Africa. Plant and animal evolution, adaptation, speciation, biodiversity hotspots. In South Africa, in the Cape region, you have a lot of endemic plants. Great East Africa, a lot of endemic fish and mollusks, etc. So there's that scope. And you can also study adaptation and speciation in these groups. Primate behavior, a study of cognitive and social evolution will give insights in how humans started off. From the earliest ancestors, is it Australopithecus, Homo habilis, Homo erectus, etc., to Homo sapiens? We can see some things reflecting in the current chimpanzees and gorillas. Adaptation to climate. I don't know whether to say in Africa the changes are drastic, and it will be a good model to use to study climate adaptation. Medical evolutionary genetics, microbiomes, these were mentioned in bees, but also in humans, these are important, and they are part of the evolutionary story. Disease resistance, also related to either the genome itself or the co-evolution with microbiomes. But instead of talking about challenges, I would talk about opportunities for African ownership of research and capacity building, including empowerment of African scholars, and also a bit of data repatriation. We have a lot of African data on African biodiversity, not necessarily here, but it can be repatriated and consolidated. And strengthening African institutional hubs, working as regional groups, or within country you can have centers of excellence, which should be funded, and the funding should include the infrastructure, consumables, personnel, etc. But also we should continue with international collaboration. We cannot overdo the independence. We should still leave room for international collaboration. Thank you for your attention. Asante sana.
Thank you, Prof. I think that was a wonderful talk on cyclic, and I would love, like, if it was for the students who are joining this aquatic science, it would be a beautiful kind of environment to get in. And I would say this is a nice one as we enter into this theme on cyclic and related studies. So this is now the time for reaction. Any questions, comments? Yes.
Yeah, thank you very much for the very impressive talk. I have many questions. Most of them I will pose hopefully over a beer or so. But one I want to pose here, that is, in the very beginning you told us of these different radiations in the 3 large lakes in Zichlitz. And if we bear in mind how young Lake Victoria is, it's actually really astonishing to find so many species. Or is there any infusion from somewhere else, or is there any explanation how that could come about in such a young lake? It was 200 species, if I recall correctly.
The latest colonization involved many species from many sources, including rivers around Lake Victoria. But I would say the primary seeding would have come maybe from the Aplochromini of Lake Tanganyika, which is speciated to some point, and then the drying caused the fish to escape. more or less like the Paleo Lake Makadikadi story. So yes, it's likely that the latest seeding would have come from multiple sources. Thank you.
Thank you. Any other? Yes, Dr.
Yeah, thank you for the good presentation. Just 2 questions. What impact could pollution be playing a role in the evolution or what you've talked about? What's going on? Is pollution playing a role? And secondly, so is there a possibility of these lakes drying up again? Should we be worried soon?
Thank you. You can respond, Prof.
Yes, I would say the pollution, there's a good paper or some good evidence on Lake Victoria. It has been polluted because of effluent going into the lake. I don't know if it's from industry or from domestic sources, but it has made the water eutrophic, which means there are a lot of nutrients, and then there are a lot of algae, then it's no longer that clear. So the fish, especially the female fish, cannot see their conspecific males very clearly. When they try to look at the color of the males, they can't differentiate between the perfect male, the perfect colored male, and the others. So this has been observed, and we are waiting for results to see whether this will cause more hybridization in the lake. But if you look at Lake Tanganyika, the water is very clear, even Lake Malawi. So I would say pollution has started affecting Lake Victoria, and those are the consequences which the female fish, or let's say the fish, especially this radiation, is facing. Yeah, in terms of possibility of dry out in the foreseeable future, yeah, I want to believe that these geological processes repeat themselves, like ice age and so on. But I don't know the periodicity, whether it's 10,000, every 10,000 years or so. And when I was researching for this talk, I also saw one report which suggests that the Rift Valley is actually moving. The tectonic plates are moving, such that in the future, Somalia and Ethiopia may become another continent. But you can't see that movement with your own experience as a human being in your lifespan. But the answer is yes, these episodes can repeat themselves, and I hope we will not have episodes like volcanoes erupting like here and so on. But such processes as drying out can happen, but very slowly.
Thank you. I know it's an exciting. I can see the time really moving. The goodness is that this is a symposium. Even when we proceed for lunch break, during tea break, we can still continue with the discussion. But if there is a burning one, yes.
Thank you, Prof, for such an extensive work on the cichlids. But in connection to Dr. Tuwei's question, I'm just wondering, how can these, the genetic diversity of the cichlids, and I tend to believe that it's one of the fishes that has wide genetic diversity, how can they be preserved in the face of the human threats to the Great Lakes? especially the African Great Lakes.
Yeah, please repeat the question.
Yeah, the preservation, how can this be preserved, the rich genetic diversity of these cichlids in the African Great Lakes? How can it be preserved in the face of the anthropogenic threats that the African Great Lakes are experiencing?
Yes, I get the question, yes. If you look at Lake Malawi, the spot where you find the very high diversity of these cichlids, the Mbuna, it's a national park. It's an aquatic park. So I think Dr. Professor Barasa and myself visited this area. You find soldiers there guarding the lake. So it is an aquatic park. It's a protected area because the country has recognized the importance of preserving those fish. And because of that, a lot of researchers are going there to do work because they know it's a protected area and there's very little anthropogenic disturbance. But for Lake Tanganyika, I think there are some efforts to map some areas of ecological importance. I think there's a project by IUCN trying to map Lake Tanganyika and identify areas of potential ecological importance which should be protected under the law of each country, each riparian country. And these are called KBAs, Key Biodiversity Areas. Coming to Lake Victoria, I don't have information on what's happening, and what I know is that there's already some pollution which has advanced, but I think it's up to the riparian countries again to come up with the programs to conserve these, and maybe it starts with you and me to make a scientific case, and then, like I said yesterday, we do a policy brief targeting the policymakers so that once they are convinced, then there could be guidelines or some legislation to protect especially hotspots within the lake. You can't exclude everyone for the sake of the fish diversity, but you can map the lake and propose some areas for such purposes. Thank you.
Thank you, thank you. I know this is a curtain raiser. The real session is coming. So we would get more opportunity. So probably on the way forward, Dr. Baraza, just one. Okay.
Thank you, Prof. In your presentation, I had somewhere you mentioned that when rivers dry up, the fish escapes to other water bodies. How does that happen? Thank you.
Yes. Let me qualify by saying when lakes dry up, usually, yeah, then the fish move to surrounding rivers. But this is a slow process. It's like the rivers, the fish have cues to detect that, yeah, something is happening, and they start to take action, and they move using their instinct. It's like that, I don't know if it's a joke, where you can kill a frog by hitting it slowly, put it in water at room temperature and start increasing the temperature. Slowly, it will not realize that it has reached a lethal temperature. So these are very slow processes. The fish starts to sense. So this is this is intelligence which we find in the natural systems. Just like termites sometimes can behave in a certain way to show you that there will be a drought. Even trees, sometimes they start to behave in a certain way. And our ancestors used to interpret some of these signs or signals. So the fish, yes, do it in good time. So once the lake is completely dry, they are already in safe environments, I would say.
Thank you, Prof. I think let us give a hearty clap for Prof for that nice insight on cyclic. We will do the 5 factorial. Yeah. Can you demonstrate for us? Kindly.
The one we did the other time with Sharon, and we had 3 factorial because the professor is here, has shown us a lot of cyclids, and we want to do 5 factorial. So we start 5, then we go to 3, 4, 3, 2, 1. So we do 5, and we do it very fast so that we go for like early. So let's start.
Thank you, Prof. So we'll break for lunch, the same venue yesterday. Then we give ourselves 2, it is now 1:30, maybe 2:15. 2:15 we should begin the next session so that we can be able to enjoy this talk on cyclic. So let us break for lunch and then we resume at 2:15. Good afternoon, everyone. So I think we can begin with the afternoon session. I hope we all had a good lunch. So the next presenter on this particular theme is P. Muhongo. Welcome and share with us this interesting area of cyclic that was Prof initiated the exciting journey. Let us enjoy this afternoon. Karibu sana.
Good afternoon, everyone. My name is Purity Muhongo. I'm a master's student from Barara University, Uganda, but I am alumnus of University of Eldoret. So, and my title today is Molecular Species Identification, Genetic Diversity and Population Structure in Kenyan Oreochromys niloticus populations. And this is just a small bit of a bigger collaborative project between University of Eldoret, Dr. James, and University of Potsdam, Prof. Raphael, and I believe Dr. Baraza will talk more about the project. So you're welcome to my presentation. So just a background. First, I would like to thank Professor Katogo. I'm sorry if I don't pronounce your name well, but I would like to thank you for the discussion that you initiated because it gave me insights of what I am actually doing. And yeah, so just a background of my title. We all know tilapia is an important fin fish economically, not just in Kenya, but Sub-Saharan Africa as a whole, it is a source of food, so it contributes to food security, as well as a source of livelihoods to farmers who practice aquaculture. So, however, there is still accurate species identification is a major challenge, even to experienced hatchery operators. This is because Oreochromis species have similar morphological characteristics. Even when we look at the pictures, we can see that these are different Oreochromis species, but you can see that when you look at them just once, you might think it's the same species, but it's really different, especially when you look at this. This is Niloticus, and then when you look at Leucosticus and Mozambicus, it's really difficult for one to differentiate these species by just looking at their morphological characteristics. And so there is need to incorporate molecular tools for species identification, because if the hatchery operators or farmers, during field sampling, they collect, they go out into the wild and collect different species, thinking it is Niloticus. They are going to affect the growth performance of the Niloticus as well as the genetic integrity of the species. Yeah, and so the objectives of this study was to perform molecular species identification of the all Niloticus species, and then after that we do genetic diversity and population structure of the population. So just an overview of the study area. So we have here Lake Victoria, Lake Turkana, and then Lake Baringo and Lake Victoria. And just as Prof had said about Great Lakes, Great African Lakes, Victoria and Turkana are part of the Great Lakes in Africa. So that's why they form an important, habitat ecologically for the tilapia species, because it's where most of the hatchery operators, farmers obtain their seeds and brood stock from. Yeah. Mm-hmm. So, and now on the methods, sampling and the lab work, a total of 73 tilapia samples were analyzed. These samples had already been, the tilapia had already been sampled. I was not the one who took part in the sampling. As I had said earlier, it is a bigger collaborative project, so Dr. Baraza, together with Prof, had did the sampling in the 3 lakes. So I just used the samples that had already been collected. So a total of 73 samples were analyzed for mitochondrial region. And then out of that, PCR was done. This is the primer that was used to amplify the control region. Cycler sequencing was done after that, and then the sequence reads were filtered and through bioedit software, and then blast analysis was done against the NCBI database to identify the species. After that, we did phylogenetic and population genetic analysis through the DNSP and Harlequin softwares. Yeah, and that's me in the lab doing some of the analysis. And then now the results for molecular species identification. When they were blasted in the NCBI database, we were able to determine that out of 73 samples, only 63 were Oreochromis niloticus. The others that were not all niloticus species were the all leucosticas and all spirulinas from Lake Baringo and Lake Victoria. And then you can see that from this total, there are two that there's two samples that didn't amplify, and so they were not included in the downstream analysis. That's why we have a total of 63 samples. And as Prof had mentioned something about hybridization that can be the results that yeah. So from the sorry, a bit test. Okay. From the results, you can see Lake Baringo studies had indicated that there is hybridization that took place where they interbred the O lesticas together with the O niloticas. So this is just to support that there are other species other than the O niloticas that are in this Lake Baringo. And then in also Lake Victoria, it's the same case, hybridization can take place. And then after a long time, after hybridization takes place for a long time, it leads to, it affects the maternal lineages. And so the native species, the genetic diversity of the native species will be reduced. Yeah. And so this one is also just to support that molecular tools provide greater accuracy in identifying the species compared to the traditional morphological approaches. And then the genetic diversity. From the results, we can see that Lake Turkana, it has the highest number of haplotypes compared to Lake Victoria and Lake Baringo, and this can be attributed to the unique ecological conditions and the relative location of Lake Turkana. Lake Turkana is quite isolated from the other two lakes, Lake Baringo and Lake Victoria, and that limits gene flow, and that's why it has like a high genetic diversity. And then also because of the ecological conditions like the fluctuating salinity and the extreme temperatures that makes the native Oreochromis become adapted to those conditions, and so retaining their genetic diversity. Yeah. And then just an illustration of still on the genetic diversity, we came up with a haplotype network, and from this you can still see that Lake Turkana has the highest genetic diversity. It has so many haplotypes, and then one central haplotype where the others are originating from. Then we can see only one haplotype was shared by Lake Baringo and Lake Turkana, and this is just also because of their location. They're all in the Great Rift Valley, and then also because of translocation that happens, especially during aquaculture species when, let's say for example in Lake Victoria where cage culture is practiced, and then when the species in the cages, the fish in the cages escape, they can move through, and then there is gene flow to the other lake. That's why we can say that there is a shared haplotype between the Baringo species and Victoria species. And so this whole haplotype networks just indicates a strong population structure of the O niloticas in the three lakes. And then we also did a maximum likelihood tree. I don't know if it's visible from your end, but we can see that there is clustering of individuals according to the population. Like here, Lake Turkana, from this haplotype to this, all these are Lake Turkana, and Lake Turkana, Lake Victoria, and then this one that is shared by Lake Victoria and Lake Turkana. And still this is just shows it just still explains the population structure of the 3 lakes. It's very diverse, so we cannot say that they have same population structure. They have distinct and different population structures between the 3 lakes. Still, this is just to support the high population structure that has been revealed. We can see that from the analysis of molecular variants, 67% of genetic diversity is due to the differences within the population, while 32% is among the populations. Even if 32 it's still a bigger number, so this still just supports the substantial genetic structuring among the populations. And then we also see the overall fixation index is still so high of 0.32. So it still just supports the diverse genetic differences between the 3 populations. And then pairwise FST analysis, we can see that, between Lake Victoria and Baringo, it's low compared to between Lake Victoria and the other 3 lakes. So still we can see that Lake Victoria is still the most genetic lake, has the highest genetic diversity among the 3 lakes. As I had explained earlier, it's mostly because of the geographical location together with the ecological conditions of Lake Turkana. And then we also, the significant, the results of the exact test population differentiation was significant. That is, the p-value was less than 0.1, very significant. So this just shows that the observed genetic differences among the populations are unlikely just due to chance. And then the conclusion, you can see that molecular identification is more superior compared to the morphological based methods, because it was able to correct the misidentified 8 samples in our total samples that we had collected. And then also you can see that a substantial genetic diversity in the 3 lakes, strong population structure also, just as I had said. And then for implications for aquaculture and conservation, we really encourage the management of the hatchery to incorporate molecular tools so as to reduce the chances of misidentification, and this improves the overall growth performance of the fish, as well as the management and the conservation. And then genetic diversity is critical for long-term population resilience, as well as the strong population structure. So it requires careful management to avoid loss of unique genetic lineages. Just as we have seen that Lake Victoria has a very high genetic diversity, and so measures should be put to conserve these native species to avoid reducing their genetic diversity. And then I would like to acknowledge my supervisors, together with all the organizations that helped in the funding of this project, and the great members from the Evolutionary Biology University of Potsdam. Thank you for listening.
Thank you, Purity, for that nice presentation. Let us respond to the presentation. Any question, any comments on Purity's work. Yes.
Okay, thank you, Purity, for a nice presentation. Mine is just a reaction, more of a consideration if it might add value to the project. I was trying to really understand the issue of haplotypes between among the three lakes, that is Lake Turkana, Baringo, and Victoria. We understand that both Baringo and Turkana, they don't have the outlets. All of them have got only the inlets, but no outlets. And then when you look at the haplotypes, Lake Turkana has got several haplotypes in comparison to the two lakes. Could it be as a result of the saline environment, or could it be connected to River Omo from Ethiopia that is draining to Lake Turkana? The other point is if we consider the gene flow, geographically, they are really separated. This is not directly to Purity. Dr. Barasa and Prof. Raf can explain. Could the issue of gene flow be considered to explain the difference in the haplotypes, having in mind that all of them are geographically apart? Thank you.
Purity, you can respond.
I don't know if I got the second one correct. You're asking that if the reason for Lake Turkana having so many haplotypes compared to Baringo, it's because of what? Limited gene of the location?
Yes. The location.
Yeah, and I thought maybe I said something close to that there's limited gene flow because Lake Turkana is very far apart compared to from Baringo. So that's why there's limited gene flow between the two, and that's why Turkana has more genetic diversity compared to Baringo. I don't know if...
Maybe just to add, he had already also asked the question of the effect of River Omo joining Lake Turkana, whether it has any effect on that diversity of Lake Turkana. Yes. Go ahead, Dr. Baraza.
Okay, thank you, Chairman. If you allow me. Certain issues here. The distance between Turkana and Baringo is large. Yeah, they are geographically separated. But if we go back to the principles Professor Katongo was bringing up in his talk, we will understand why there are shared haplotypes between those two lakes. There is what is called historical connectivity, and then when dry conditions came, the connection dried up, and because of tectonic activity, there was that separation. But otherwise, historically, there was a connection between Lake Turkana and Lake Baringo, and the connection was by a river called Kangwen. Yeah, there was a Kangwen River. If you read some of the old books and literature, you get that name. And it explains why there are shared haplotypes between those two lakes. Now, for Omo, Omo, the impact of Omo might not be so much because Omo ends in Lake Turkana. Yeah. Omo is the main inlet for Lake Turkana, and its influence ends in the lake, in Lake Turkana. Yeah. After that historical event, there is virtually no connection between the two. And if you look at Turkana, aquaculture around it is also limited, and that's why if you look at the network, you would see some distinct structuring for Lake Turkana. For Lake Baringo, it could be almost similar, but we know that Lake Baringo, the catchment has heavily been impacted by aquaculture because of irrigation activities in the Akera scheme. And also, Baringo County is also, over the last decade, has been investing heavily in aquaculture, especially in the upper part. And so there is some influx from ponds, from reservoirs into the lake. And that also goes back to explain our results from the first objective, why Lake Baringo has several species. Thank you.
Okay, thank you.
Yeah, Prof wants to add something.
May I add a comment? And I think Purity did a very good job in presenting us these data. If you have a look at this haplotype network and just put kind of a virtual line above the number 2, the red big one, then it's almost on the one side you have only Lake Victoria and Lake Baringo, and on the other you have mostly Lake Turkana, with the 2 exceptions of Lake Baringo. And that actually tells us that there's, for quite a longer time, different evolutionary processes, not completely separate. And I think you also asked why there are so many types in Turkana. That could indeed mean that that there is a relatively high population size of fish, and the influence of other. James already said Omo may not have a large influence, but as we don't have reference samples included from other areas, we cannot really say how it relates to other areas. But one can already see there's quite an independent process, evolutionary process in Lake Turkana. While apparently from what James said, Baringo may have been impacted by this aquaculture method, because Baringo has not really a clear pattern. There's one haplotype here, one there. So.
Thank you, Doctor and Prof.
There's another question here.
Thank you for the nice presentation. I'm just wondering aloud, if the two lakes are far apart, epigenetics takes control in terms of diversity, and then when we're doing our molecular analysis, you opted for mitochondrial DNA.
Please speak up a bit.
You opted for the mitochondrial DNA. Mm-hmm. And not the genomic DNA. What is so special about the mitochondrial DNA and this role in epigenetic regulation of diversity across the 2 environments? Thank you.
The reason why I opted for mitochondrial DNA, not whole genome DNA, whole genome analysis.
I think the question is, why did you use mitochondrial DNA and not the nuclear DNA in your analysis?
Because we know that mitochondrial DNA is the one that controls the maternal lineages, and that's why I opted for that.
Yeah, in fact, we have 30 genomes of these fish as well, but it was beyond what Purity could achieve in her several months stay in Germany. So the analysis of the genomes is underway, but this was mostly thought to make the species identification for which the mitochondrial DNA is very good, but it already tells us an interesting story about the structure as well. Yeah. But the genomic analysis are to come.
Thank you, Prof. Another question here?
Yeah, thank you for the good presentation. My concern is about the impact of the results of the study. And I'm wondering, as a layman who is not into fish, or maybe who is not into the whole thing, what is the take home for us? Because I'm thinking in terms of conservation, maybe the data should be communicating something. That's one. Number two, I'm wondering if the larger study considered checking on the environmental factors that could influence the diversity, because that is where the message I'm interested in comes from. So I'm wondering if you're doing that, you're checking the condition of the waters and the things around there that may affect the diversity of the different fishes in the lakes. Thank you.
Okay, first, the impact of the results to a layman, in layman's language, to people who don't understand maybe genetic work. That's why we have something called like integrated management, integrated natural resource management, where we have to incorporate so many people into the management. We don't just communicate to you people only. We have to, like, just the way Professor Kinyua said something about putting all of us together in a room and then explaining to them what is supposed to be done in terms of policies also. So we have to incorporate so many people in and explain, because for us, maybe we're just only interested in the genetic part, but then there's also the part of maybe the government in terms of policies related to management and conservation. So we have to, like, come all of us together and communicate these findings to everyone. So we have to incorporate the community as well, the farmers, the hatchery operators, the government, the genesis scientists, and discuss all this. Yes. And then, sorry, maybe I lost you on the second part.
The environmental, I think there was something on environmental impact.
How the environmental impact affects the diversity?
Yeah.
I don't know if I discussed something like that, but I just thought maybe the high diversity in Lake Turkana is because of the environmental conditions. Yes, the high temperatures, it affects diversity, because the species in that lake will be adapted to that environment over time, local adaptation, and then that will impact the diversity of that lake. I don't know if...
So I think from the layman point of view, the question is that good or is that bad? That is what you would want to understand.
It is good, because you can see that now it has made the genetic diversity of Lake Turkana to be very high, and that is what we want. Yeah. So that the native species in Lake Turkana can be conserved, so that they don't disappear. Like, for example, when hybridization took place in Baringo and Victoria, that over time it led to some native genes being disappearing.
So there's another question, maybe a follow-up on the same.
But yeah, so it relates to the difference in the genetic diversity in the Turkana and Baringo. These two lakes are also quite different with respect to their size. So you can imagine that in Turkana there's a higher, like, census population size, yes, which might also translate into a higher effective population size. Yes. That can maintain much more diversity, and there's maybe more genetic drift in Baringo, which is rather small compared to Turkana. Yes. So how is that a confounding factor that is difficult to control in your setup, or does this relate to this? I'm not a fish person. I have no idea whether that is correct what I'm saying.
Yeah, you can respond to that.
Yeah, that is okay. Like it has been confirmed, so it's fine. Okay.
Thank you so much, Purity. I am not so much into this, but I see this thing has attracted many of us. Maybe what I can say is that you need to document other factors very well, so that maybe when you're presenting, they sound high, and we don't have to ask for them. Because if you may realize, it's like we are digging into your work.
Yeah.
Make sure you let us know other issues that you surely considered. So once you can have those on paper, and maybe any other person can read them, they may introduce some of these other inquiries that are coming up.
Thank
Otherwise, good work.
Thank you.
Thank you.
Thank you for that comment. Prof was having just a word to add.
He has decided that I was going to add a word. So my word is this: purity. Well done. The presentation is okay. So my word is a question, which it now turns out to be a follow-up. The other factors that you need to look for that are bringing out the diversity, because even though Dr. Barasa talked about the historical connectivity, it didn't come out. So I think that one died long time ago in the history. So you need to look for the other factors that are that are bringing up that diversity. But now my question was, why did you need to compare the relatedness in the species in the 3 lakes? I was hoping that Dr. Barasa has answered that question, but you've spoiled it by bringing up other factors. So why, I mean, what did you want to prove? What was your hypothesis? What were you following up so that you should compare the 3 of them? Because of course, for you to follow up, it's because you're being guided by something. I mean, what is that? That's what I've been trying to work out. He has talked about many factors. Yeah, that's true. But now those factors, even after digging, even after forcing her to bring them out, for me, it hasn't yet come out what guided that wanting to compare the 3 lakes.
Yeah, so you can respond to Prof.
Okay. I had said earlier that the 3 lakes, from the 3 lakes, we have 2 that are great African lakes, and they are of importance because it is from these lakes that we get the broodstock for the for hatcheries, as well as the seeds, and so these are among the 3 major lakes in Kenya. So they have different ecological
So are you wanting to prove that the 3 very important lakes have got diverse fish, and therefore when we want to do something,
yes,
whatever we want to follow the lake that has got that diverse?
Yes, I wanted to prove that the 3 lakes are distinct and they have like different diversity. Yeah. Yes.
Yeah, Prof, you can add.
On that, and I think actually it was already on one of the later transparencies of purity, but I think if we take the layman perspective, we can actually draw 2 conclusions from this. One is for aquaculture, there's a lot of diversity in the Kenyan tilapia, which is good. But for conservation, don't mess these strains up. I would draw that conclusion. Don't make sure that you don't bring fish from Turkana to Victoria or like vice versa, unless you don't care about the local adaptations. So, and I think you spelled out these two messages also on one of your last transparencies, but that would perhaps an easy take-home message.
Thank you for that response. Do we have any other? Daktari?
So mine is an observation, and that is triggered by the high genetic diversity of fish in Lake Turkana. I think there is something unique about that region, because archaeological evidence shows that even the early man kind of lived there, because the work of Leakey, the archaeology done in around Opo Valley, I think there's something unique about that. I think we need to factor in. I just want to trigger curiosity about that entire region. There is something quite unique about it, and this adds to it. So I think people should start thinking, what is so unique about that place?
Yeah, yes.
Thank you, Daktari, for that input, and I think this goes back to what Prof. Katongo was mentioning. There are a lot of opportunities looking at this particular fish speciation, such that there are very many openings that could be emerging from these particular studies, related even to human development, related to how these fish have developed. These kind of opportunities are quite many, and I think this collaborative kind of work that is going on could be able probably to reveal some of these things coming up. I don't know whether you can add something?
No.
There is one burning from behind. I think it's an interesting area that is attracting a lot of attention.
Yeah, it seems so.
Thank you. For me, I'm still curious. I want to understand the great diversity that exists in Lake Turkana in comparison to the other lakes. So, I'm asking myself, could it be attributed to economic activities that are taking place within the surroundings? Or could it be attributed to physical conditions of the water within the lakes? Or could it be attributed to microbiological conditions of those lakes? Or the historical basis for the same?
I feel like all the reasons you have mentioned could attribute to the high genetic diversity in Lake Turkana. We can see clearly there's a lot of, everything is going on in Lake Turkana, so there are so many reasons that can be attributed to that. Mm-hmm. Yes.
The audience wants to hear what you're saying.
Maybe we should discuss this over tea with purity. Because, Lake Turkana has got, I teach genetic diversity. That's what I teach about conservation and Lake Turkana is less disturbed than Lake Victoria and Lake Baringo. Yes. Lake Baringo and Lake Victoria are more disturbed, and that means that the diversity in Lake Turkana is more, that's why it is more than the others. Because when you disturb by selection, by biased harvesting, you know, all those. And that can affect the diversity, rather than even what he has talked about.
Dr. Baraza wants to add?
Yeah, so I'll just add, ideally most of the things that are contributing to this have been mentioned. One is the large size of the lake and therefore of the population. Then less disturbance, like Professor said. Then next is the environment, the good environment. Very limited impact. That weather, the high temperatures favor this. How about exploitation pressure? Exploitation pressure in Lake Turkana is also very low compared to these other lakes, so that adds. Then, perhaps even more important is that there is also, the Lake Turkana is also connected historically to the Nile province. Yeah, the Nilo-Sudanian province. Yeah, you know this, Purity. And that province, that region, ichthyological region, is highly diverse. And these are some of the things that the professor was talking about when this sub-theme was being introduced. So a range of factors are contributing to Lake Turkana, showing that kind of diversity. So you just need to put this into perspective and bring them out clearly. Thank you.
Thank you, Purity. I think this is a symposium. We can continue this discussion over a cup of tea, over dinner, so that we give an opportunity to the other presenters. Let's give Purity a good hand clap. So I want to welcome the next presenter, Daisy. Karibu Daisy for the presentation on towards conservation of endangered Singida tilapia. This is again another very interesting study. Karibu sana Daisy.
Thank you so much. My name is Daisy Nafula Simiyu. I'm an MSc student doing fisheries and aquatic sciences at the University of Eldoret. And maybe as we start, I'd like to highlight the fact that my research is ongoing. We have started, but we are yet to make conclusive, to get conclusive results. So I will be presenting basically my proposal and what was the thought behind this particular study. So, as you've seen, the topic is towards conservation of endangered Singida tilapia, that is Oreochromis esculentus, in the Lake Victoria Basin. Sorry. So, Oreochromis esculentus, or Singida tilapia, is locally known in on the Kenyan side as Singege tilapia, and it is a species on the brink of extinction, whereby in the previous years it formed a very important commercial and commercial catches within the lake basin. And just a point to note that it's found not only on the Kenyan side of the Lake Victoria Basin, but also in Uganda and Tanzania. So according to the IUCN Red List that was in 2021, it is critically endangered and on the red list. And you can see they have put there 2026 because as of 2026, it's still on the red list. And we find that the remnant populations now only persist in isolated small water bodies and the satellite lakes of the main Lake Victoria. This is because when you look at the satellite lakes, that is Lake Kanyaboli and Lake Namboyo, where the species is found, you will realize that they are separated, completely separated from the main lake, and therefore this species still exists in those areas. Looking at the decline of the species from the lake basin, we see that in 1900s up to around 1920, it was in peak abundance, and that, yes. As we move towards the 1940s, we find that overfishing started reducing the populations. This is because there was introduction of more efficient and effective fishing gears. So the population had started increasing. Towards 1958, we see that that is the point at which we started seeing the introduction of the other tilapines. And as we move towards the 1960s, we see that the populations of the main, the genetically pure, if I can call that, has started decreasing. And when you get to around 1980, the population had decreased from the main. When we talk about the population, we are talking about the genetically pure populations had started disappearing. And by 2000, that is post 2000, we find that now that is the point at which they were declared to be critically endangered. However, they could only be found in satellite lakes. So what are the causes of this decline? We find that environmental degradation has led to the loss of diatoms, which was their main food item. We also see that the exotic species that were introduced, such as the Oreochromis niloticus, they hybridization. You look at introduction of Lattes niloticus. It is a piscivorous species, so we see that there was a lot of predation from that end. And also from the exotic species, we see that there was also competition for resources. We move to another cause of the decline is anthropogenic activities, whereby basically the agricultural activities, we find that they are leading to runoff that gets to the water, therefore affecting the water quality, and overfishing, as I had previously explained. Thank you. For me to be able to come up with this study, we were able to look at various previous studies that were done on the species, particularly within the lake basin. So we look at Agnes et al. In 1999, in the paper in 1999. They studied the Lake Victoria, the main Lake Victoria. They used allozymes as well as the microsatellite DNA. And from there, they were able to determine that there was genetic erosion and hybridization due to the availability of mixed microsatellite alleles and altered allozyme frequencies, meaning that there was integration within the lake. And therefore, among the findings that they were able to come up with, that the populations within the lake were highly hybridized and suggested that isolated populations, there was not much hybridization. We come to Angienda et al. In 2011, their paper in 2011. They were able to do their research in Lake Kanyaboli and the surrounding wetlands. And they used microsatellite DNA, the mtDNA, and clustering. From there, we see that statistical clustering, it was able to group the individuals genetically and estimate the levels of admixture between the species. And from their finding, we see that they were able to see that there was low introgression between Oreochromis esculentus and the Oreochromis niloticus and Oreochromis variabilis that was found within the Lake Kanyaboli. Angienda and et al. Were also able to identify unique haplotypes, 3 unique haplotypes to Lake Kanyaboli population. They were also able to identify 4 unique haplotypes, that is private haplotypes to Lake Namboyo, and 1 shared haplotype between the two satellite lakes. And therefore, the conservation implications of their research was that they were able to identify satellite lakes as genetic refugia. When we look at the studies by W.W. Mwanja et al., that is between 1996 and 2012, the studies were done on the Ugandan side of Lake Victoria, and they used ecological surveys, morphometrics, and molecular genetics, whereby they were able to identify that there were declines of from the main lake. That is decline in terms of numbers. They were also able to identify that hybridization occurred within the lake, and that pure populations had completely reduced. And but they were also able to identify that pure populations were found within this satellite lake and other small water bodies of the Lake Victoria and Lake Kyoga systems. So, what was the problem a research problem that we were able to identify? We looked at it that there was a critical knowledge gap, whereby there's no contemporary integrative data on the biological characteristics and environmental suitability of the small water bodies for the persistence of this particular species. And we thought that understanding the historical introgression and genetic diversity as well as hybridization is important for coming up with species-specific breeding protocols and site-specific conservation intervention. And because historically this species was important to the fisheries of the lake basin, we thought that restoring the population will support high niche markets as well as contribute to food security of the lake basin. The study objective is, again, as I said, this is part of a larger project. We are presenting the genetic aspect, however, it is a larger project. So the study objective is to investigate some of the important biological characteristics and environmental influences of this particular species, that is Oreochromis esculentus. And the specific objective for today is to determine the genetic diversity and population structure within the lake basin. Sorry. So for our study site, we were looking at 2 of the satellite lakes of Lake Victoria, that is Lake Kanyaboli and Lake Namboyo. We're also looking at some of the water bodies, small water bodies, whereby this species has been identified and noted. That is Gesebei Dam in Nyamira County. And we have Kalenjuok and Nyalnawe Dam in Siaya County. So the reason as to why we were looking at these small water bodies, it's because we wanted to see if there's any difference between the genetic makeup of the species of the fish that are actually found within these small water bodies and the satellite lakes, and we also want to see if there's any difference within the population structure. So as you can see, the small dots are representing the areas where they are found. So you look at Gesebei Dam, you can see geographically it's a little bit far away from these other areas. So the methodology, we did, we have done one field sampling, but we hope to be able to do the second field sampling activity. And as I had said again, this is a part of a larger project whereby we will be able to, among the measurements that will be done, the in situ measurements will do the water quality. We'll also be able to get the GPS. While in the field, we'll also be able, we were able to also get the length weight of the fish, the length and the total length and the total weight. We were also able to get the stomach contents that we were able to preserve for lab analysis. We were able to get the gonads for the female fish, that is to be specific, for us to be able to get the fecundity and the GSI, that is the gonadosomatic index. And most importantly, we were able to get fin clips that was preserved in 95% ethanol for DNA analysis. Other than that, we were able to talk to the community around and get the indigenous knowledge. We were able to talk to the fishermen. We were able to talk to the fisheries officers and other conservationists within the study area. So we took 35 fish samples. This is to ensure that there was a representation of the diversity within and among populations. And as I said, we collected the fin clips and stored in 95% ethanol. And as at now, we were done with DNA extraction. We are still waiting for, we will be commencing on the PCR and the subsequent processes in the near future. So this is my colleague Lamek. He was here with us. He's also part of the project. Here we were taking the in situ measurements, and here we were identifying, identifying the species that we wanted. That is, we were using the local community as well as a fish taxonomist from, that is the Kenya Marine and Fisheries Research Institute. Here we were doing the, this is me. I was doing the DNA extraction, and this one is Dr. Baraza. After doing the extraction, we had to go and read to ensure that we have actually extracted something from, gotten some DNA. So the aim of this research is we are hoping to reinforce the information that satellite lakes and other small water bodies are important for conserving the native tilapia biodiversity. We also want to reinforce and insist that molecular genetics is essential for identifying the pure remnant populations. We also want to reinforce the fact that conservation interventions are urgently needed to ensure that Oreochromis esculentus continues to persist within the ecosystems, and that scientific basis for conservation breeding, genetic monitoring, and protection of wetlands is important in fisheries management. So this study will, for us to be able to get the aquaculture potential of this particular species, we are adopting a multidisciplinary approach whereby we are using population genetics as well as conservation biology, whereby we are understanding the environment, the population structure, and the threats to be able to identify the potential and the aquaculture potential of this species. So what are our expected outcomes? We expect that at the end of it all, we'll be able to identify environmentally stable habitats with favorable physicochemical conditions and diatom availability for conservation. We also hope to be able to get to identify populations with superior genetic makeup, fecundity growth, and reproductive performance for species-specific hatchery development. We also we expect to develop species-specific ex situ protocols for captive breeding and the potential of reintroduction to the main lake. And lastly, we hope to be able to provide evidence-based guidance for habitat protection, seasonal fishing closures, spawning ground designation and protection, and basically the entire ecosystem and biodiversity conservation. So as I finish up, I would like to acknowledge my supervisors, that is Professor Philip Raburu of the University of Eldoret, Dr. James Baraza of the University of Eldoret, Lamek Otieno, who is our assistant. And we have Edith Kurui, who is my lab partner. She's also working, she's doing her PhD, and she's also working on genetics, so we try to share ideas. And lastly, Professor Ralph and the team from the University of Potsdam. Last year in September, they were able to take us through training on bioinformatics, which I do know that will be very useful when I'll be doing, when I'll be done with this and doing my analysis. Thank you so much. And
thank you, Daisy. Let's give her a good clap. So, time for reactions. I think Daisy would be lucky to get some useful comments as she proceeds with this work. So let's start from the back.
Thank you, Daisy, for a good presentation. My question is on, I'm Sarah Waswa, an MSc student. My question is on species identification of the fish. You've mentioned that you are going to utilize a fish taxonomist to help you identify the species of the fish that you are capturing, yeah? So which metrics are you going to use to be able to confidently call that species? Because you know there are some studies that I've seen some studies when they're taking those species of fish, they dissect. I don't know what they look at. I'm not that much familiar with those metrics, but I would like to know how confident you are with your morphological identification of the species, and what would happen in an instance whereby you get some discordance. One person is calling this species this, and another person is calling it another. Okay, you might not have two different people, but in my case, I had a discordance between different species, and we had to choose one strategy over the other. So that will be very interesting to see from your point of view.
Okay, thank you so much. That's a very good question. And from the experience of, as I said, we were using not only the fish taxonomist, but we were also using the in the fishermen and the individuals who have been within the community. So, and again, remember we said we are using 35, we are taking 35 samples. So from their experience, we were able to correctly identify, using their, that knowledge that they have, we're able to use that. And wherever there are any, what are they called? There's any disconnect, then we were able to move to the next species. Thank you. But maybe as I said, this is a continuing study, so that is something that I will note down, so that for future we will be able to, avoid any disparities. Thank you.
Yeah, just a follow-up comment on that. You can't move from one species to the other if you find a discordance because this is real science. You are going to disseminate this knowledge to the world. So when it comes to that, there are some type of strategies that are employed to curb that. I would like, or rather, I would just like to say that there are always two different, or rather more than one solution to a problem. You can always look at multiple solutions.
Thank you so much. And maybe just to add on that, I think there are also identification keys that can be used. So thank you for that comment, and it's noted.
Thank you. Thank you. Any other?
Thank you, Tay, for that nice presentation. I believe on the issue of identification, whatever you're doing in the field is presumptive, and when you are doing molecular, you are doing confirmatory identification. And then I have two questions. One, you are talking of identifying a stable habitat for rearing of the fish. But in your presentation, I never heard of how you are going to study the different environments so that you can conclude maybe this is the right environment or this is the alternative way to keep or to rear the fish. And then the second question is on the issue of identifying fish with eyes. Parameters are you going to factor in that will guide you into achieving that objective?
Daisy.
Thank you so much, Ezra. As I said, this is a part of a larger project, and we are actually working on 4 objectives. And one of the objectives is environmental suitability, but because this is a genetics symposium, we were able to only present the aspect of genetics. But as you can see among the key measurements here, we also have something on water quality, the plankton availability, the chlorophyll A, and other nutrients. I think that's okay.
Let's give Naman.
Yes, I did not actually get, thank you. First of all, I didn't get your study hypothesis, and I also saw that your study will begin from maybe ending 2000 onwards, towards the extinction of this particular ngege. There was this Mbita Causeway, that man-made blocked fish from coming from Kenya to Uganda. And that might have also contributed to the species extinction. Now that blockage is no longer there, and the fish is able to migrate. Are you sure that we are going to realize our hypothesis, or we are going to omit type 1 error? And then, some of these fish are migratory. With opening up and maybe that barrier at Mbita Causeway, and maybe there is more food along the River Nile towards Cairo, maybe that might have contributed to maybe the population moving along the River Nile towards Mediterranean Sea. Again, the settlements in these islands in Lake Victoria, like Migingo, why is the war? That fish population is perhaps too many in this side of the lake and not the other side. And what is the impact of breaking up of the man-made causeway bridge that was there before? Thank you.
This probably you can respond as you respond. I was also wondering aloud whether Lake Victoria is part of your sample, or in Lake Victoria is completely extinct, so you can't get any of these ngege from that particular lake.
Maybe I can start with answering your question, but thank you so much for your questions. And according to the study by Angenda in 2011, the fish, the Oreochromis esculentus in Lake Victoria, it is completely hybridized, so we are not able to get pure populations. That is from their study. So we are building on the study from the previous. That is why I did an analysis. Like we tried to check on the previous works that had been done before we were able to come up with the areas of study. And I don't know if Naman, I got your questions, but you are asking about the implication of the populations in Lake Victoria, or what? Thank you so much, Naman. I think I've gotten you. However, based on my area of study, if I understand well, Mbita is in Homa Bay County. And maybe that's something that I can look into later on. But currently, my study is based in Siaya County, which is a little bit far away from Mbita County. But that is something, thank you so much. That is something that maybe I'll have to look into.
Yeah, thank you also from my side, Daisy, for the nice presentation. It reminds me, your presentation reminds me in a way to our first keynote from Paul Grobler, that you have a rare species and a common brought together, and then the rare one is actually affected by potential integration and hybridization, which happens in Lake Victoria. That said, if you look at the other small lakes, you kind of assume or hope that they are pure, but with mitochondrial DNA, you have a maternal marker, so you might have issues in detecting whether they are really pure or not. So I wonder whether you aim at including also microsatellites or other nuclear markers, and if you're interested, I can also give you some hints on that, because that is, of course, from a conservation perspective, you would to know, I guess, whether these satellite lake populations are really pure or not.
Thank you so much, Prof. I think that is something that I will be interested in the future. But for this study, maybe this is where we are best. But it's something that I'll be interested in. Thank you so much.
Thank you, Daisy. I think we have really tried to get Daisy moving in the right direction. So if there is no banning, we can allow Daisy to give way to another presenter. One.
Yeah, I don't want to qualify it to critical unless this August House says so. Thank you so much for the presentation. You also resemble one of my friends in Uganda. I thought I should also say that. Now, mine goes to the citations. There is some table you displayed, and you gave us 3 citations. But out of the 3, it is only one that was valid. And it is critical because you've brought this for public consumption. So we might need to, that if you are citing, complete it. Yeah, that's very, I called it minor, but it may end up being critical. Then, we will also be happy to see in the near future, like you've promised us, that we see how you are connecting conservation to genetics of the fish. We will surely be happy. 3, it goes back to what the other madam talked about in the corner. We will be also happy to see that you've stated well or documented the identification procedure, so that we don't have to ask or maybe say this and that about the procedure. So it is good that you document it, so that whoever reads this work can go through the procedure and appreciate. I think that is all I can say. Bless you, Sereko. Thank you.
Thank you so much, Blasio. It is well noted.
Thank you. Let's give Daisy a clap. The next presenter can take the floor. Dr. James, welcome to your presentation. I think you can 15 minutes to be able to present your work, and then another probably 10 minutes to interact.
So thank you. Thank you. So my talk is on the evolution of the sex-determining locus in natural and farmed tilapias. The talk is weaved around some of the work that we've been doing and is still ongoing. But I will just give some highlights on the evolutionary aspects. I'm doing this together with Professor Ralph and Dr. Marisol, who have been part of the project in the initial stages. To moving. Okay. So tilapias generally look like that. You've been seeing these photos since mid-morning. Obviously, they are good specimens. They are nutritious and palatable, and they have a global appeal, a global demand because of the it's consumed globally. On farms, they have fairly high growth rates. They survive in high numbers. They have high fecundity and, of course, excellent meat quality. And these are characteristics that are very important to the farmer. Globally, that is the production data from farms. Of course, China is leading. And as you can see, most of the lead producers of farmed tilapia are in Asia and partly in South America. Of course, from Africa, we have only two. Okay, Uganda and Ghana are in that league, but of course you can see that their quantities, their tonnages are not substantial, but they are our lead producers. And nevertheless, despite that kind of situation where Asian countries are lead producers, they produce using germplasm from Africa. Tilapia is naturally germplasm or natives of Africa. And as you can see from that map, there are about 7 native species on the African continent. So except the Middle East, countries like Israel, most of the countries that produce tilapia use germplasm from Africa, despite the fact that Africans ourselves, we are producing very little. Generally, tilapias on farms are grown in different systems, and this is just, these are cages. These are ponds where you introduce net hoppers, and rich farmers or fairly resource-endowed farmers use raceways under greenhouses, and this is a trend towards intensification. But these systems generally face challenges because of issues related to sex, because sex as a trait in farmed tilapias is very important and always very difficult to control. So globally, economic production of tilapia relies on the use of all-male seed, and our project revolves around this. Why all male? Because males grow faster naturally. They have a natural ability to grow faster. But additionally, farmers who mix sexes incur losses because of slower growth. Why slower growth? Because tilapias on farms are also prolific breeders, and so they convert energy that should go into muscle building into excessive reproduction, in which case we end up with very many young fish of low market value. So it becomes uneconomical. So in Lake Victoria, this is generally what you would see Yeah, there is massive investment in cages, cage culture of tilapias. On the Kenyan side, we have about 6,000 cages. This is a very lucrative enterprise, and many farmers are going into this. Why is this important to us? Because all these cages need all-male seed. They should be stocked with all-male seed. But I've already said that sex is a challenge. So you find that most seed that is stocked here is not all-male. There is a mix. So we really need to address this, because if there is a mix, then it means there is a reproduction in the cages. And with the reproduction in the cages, the fry will escape into the open waters, and then they will interact in negative ways with the natural tilapias that are there. Currently, methods of producing all-male tilapia seed are complex. Farmers use this but each of these is complex or laborious or inefficient. So our project is really geared around finding a more efficient approach towards producing all-male tilapia seed to support intensive cage culture on the lake. Generally, these methods produce about the success rate is just about 70 to 90%. So you can see that that is a problem. So currently, genomic approaches allow us to approach this challenge differently and perhaps more efficiently. And so I'm just going to give a highlight of evolutionary trends for this important trait of sex in tilapias. But tilapia sex determination, genetic tilapia sex determination, is premised on the fact that the different species have different mechanisms. So there is one group where we have heterogametic males. That's the first system. But the second system, we have heterogametic female system. This is expressed in different species, and the larger project is geared towards exploiting these differences and these evolutionary trends. So for Oreochromis niloticus, which is a native of the Nile Sudanian province, you find that sex-determining locus is located on the lineage group number 1, for both female and male. And these studies were done way back in the 90s using microsatellites. This is perhaps the most widely farmed species, and you find that its sex-determining locus is located on lineage group 1. But if you look at a different species, Oreochromis aureus, which is the blue tilapia, you find that the lineage group is either on lineage, the sex-determining locus is either on lineage group 3 or lineage group 1. So this is interesting, because then what drives this? For Lake Tanganyika tilapia, you find that the sex-determining locus is also located on lineage group 3, as shown. So you can see that some species have similar evolutionary trends for sex, but some show some variations, and these variations are very interesting from the evolutionary angle. So these are some of the examples of the species. Some of the species, you find that the O. aureus from Israel will show this. But O. aureus, the Egyptian strain, will show both loci. This is rather interesting. if you look at O. mozambicus, it has both linkage group 1 and linkage group 3, which is similar to O. aureus from Egypt. But you will find that if you look at a different strain of O. mozambicus, probably the linkage group would be different. So these are the interesting trends that we are interested to exploit. So for tilapia zili, for example, we see that the linkage group is, the sex-determining locus is on linkage group 1, and you can see that there are variations here, which if we understood properly, we would find an easier way of approaching the challenge of producing all-male tilapia fry for use by farmers. So these results show that there is considerable plasticity in sex determination in tilapia species and strains. Depending on the species and the strain, you find variabilities. So it is very interesting to know why the locus will vary from species to strain and vice versa. So variation depends on the species, strain, family, and some minor factors, or what is generally called the autosomal locus. The autosomal locus is one that is not related to sex. And these are some of the issues that are of interest to investigate in the larger project. So some of the unexplored questions. We've seen results from different species and different locations. So our interest is on the Kenyan strains and Kenyan species. What would be the expression for the Kenyan tilapias? And of course, in Kenya, we also have several species. But our focus is, of course, on Nile tilapia, or Oreochromis niloticus, which is the most widely farmed species. So our larger study is geared towards these questions. But apart from this, also there is a general understanding that tilapia sex determination is also influenced by environmental factors, and the most important environmental factor is temperature. So what happens if you rear your tilapia at the hatchery at slightly higher temperatures? Naturally, it's known that elevated temperatures of rearing tilapia fry would lead to a higher percentage of the fry being males. So we want to exploit this understanding to see what happens now under temperature-sensitive locus. This temperature-sensitive locus for sex determination, on which linkage group is it located. So these are some of our approaches that we will be investigating at the hatchery. And it's geared around these 3 lakes, which Purity has already talked about. Lake Turkana, Lake Victoria, and Lake Baringo in the Rift Valley. We've already done field sampling at Lake Turkana, and you can see that we could get the fry, and that's the field expedition. So generally, this is just an overview of what we are doing and the relationship from the evolutionary perspective for sex determination. Thank you. This is how tilapias look like. They are very delicious. Thank you.
Thank you, Dr. Ari, for that insightful. Let's now react to this particular talk. Prof.
Thank you, James. Excellent presentation. I'm not so updated on tilapia overall taxonomy, but it's very striking that you have in the same group these two totally different systems for sex determination. So if one looks at phylogenies of tilapias using other markers distributed across the genome, is this something that dominates the whole picture, or is it not so significant? In other words, the split between the two types of sex determination systems, is that dominant in the overall tilapia classification, or does it not play such a big role?
It is what I highlighted was specific to particular species, but so the variation is related to species. From the phylogenetic aspect, I'm not sure if you will be able to figure out the patterns. Yeah, the patterns. I'm not sure.
Well, it will be very interesting for somebody to look at that.
Yeah, I think so. Yeah.
Yeah, thanks. Part of that is evidently could be at least potentially solved by whole genome analysis. And indeed, as I mentioned previously, we have now 30 genomes, 5 of each sex and all 3 legs. As Purity showed us, there's profound population structure. That, of course, kind of confounds the search for the markers. So, to the best of my knowledge, what James also convincingly laid out, there are the linkage groups known, and we have indication that linkage group 3 is also involved in the Kenyan strains. That said, to finding the exact locus, it's still changeless. People have found associated or linked SNPs, but they are prone to differences among populations. They may be linked to the sex determining locus in one strain, but not in the other. So, yeah, that is a challenge to find that. We are on that. We have to see whether 30 genomes is enough, but that is definitely the way we need to go.
Thank you. Thank you.
There is another question at the back.
I have a question, Dr. Barasa. In the era of artificial intelligence, what will be your opinion on using mathematical modeling and statistical modeling in identification of the sex-determining locus?
Yeah, I think that would be an interesting approach, and perhaps it will fasten some of the patterns that we need to understand before we make some considerable progress. But again, we always have challenges with the models and the applicability. Yeah, but it's something that can be explored to improve the insights that we need to see.
There's another question from Ezra.
Yes, and thank you, Daktari. I have 2 issues that have been lingering in my mind. One, could you develop a system that could forecast a change in either the pond conditions or the lake conditions that could help in monitoring the survival of fish, the tilapia, since we understand that with change in climate could result to rising temperatures of the water bodies, which directly affects the sex locus. Could you develop, since you're working on the same, could you develop a system that could forecast? Because they will be relaying real-time data on the changes, on the environmental changes occurring within the water body that we rear our fish. Then the other one is the issue of male growing faster than the females. Could we employ the gene editing technology and we also edit the one for the females so that they could grow at the same rate with the males?
Okay.
Yes. And then related to it is, could we say that those who are fast growers mature faster than the other ones?
Can respond, Dr.
Okay, thank you, Ezra. Well, for the models to address temperature, we are also keen on the optimal temperature, because there is a limit beyond which the fish will die, whether they are in ponds or in the natural environment. So yes, you can do this in ponds, but of course, if you raise it beyond a certain optimal level, then high mortality will occur. That's why it is perhaps inapplicable, because we have to be sensitive to the fact that the fish will also not survive beyond a certain temperature. What was your second? I didn't. The second question.
The second, it wasn't more of a question. It was a concern that could you develop, since you will have studied all the necessary conditions for the evolution of this sex-determining locus. Could you develop a forecasting system that would be responsible for monitoring the environmental conditions, whether in water bodies or in ponds, as a warning in case of any changes that could alter the sex-determining locus of a fish?
I think it's related to what I've just said, because there is an optimal temperature beyond which the fish will not survive. So if you are introducing this to the experiments you will set at the hatchery, perhaps it's possible. But there is also an optimal temperature, even if when you are doing it at the hatchery, you want to rear the eggs or the fry at. If you exceed that temperature, then the survival becomes very low. Most of the fish would die.
Thank you, Dr. Ari, for that. I want us to transition. I hope we have to understand, and my watch is telling me it's already past 4. We still have one other candidate for presentation in this particular theme before we exit to the next session. So I want to allow discussion to go over tea in the evening, then we allow, there is a Mandua, unless somebody has a really burning. We want to transition to the next presenter. Thank you, Dr. Let's give Dr. Baraza a clap. So I don't know whether the next presenter is ready. Mandua.
Yes, I'm ready. Can you hear me?
Yeah, we can hear you very well. You have around 15 minutes to present, and then probably 10 minutes to interact. So the floor is yours.
Okay, thank you. All right, so I will transition back to, I will turn off my video if the network is bad for me, but I am Sula from the University of Malawi. I am presenting on DNA metabolic coding for Lake Malawi fishes. So my study is for my MSc at the University. environmental DNA is, so I'll just start from the beginning. Forgive me if you already are aware of this concept. But for environmental DNA, most of the times fish will release some parts of themselves in the environment where they are. In the case of fish, it means in the water where they are. So it will be in the form of fecal matter or mucus or whatever, even when they lay their eggs and everything else. So you can get the presence of a species based on their eDNA without actually having to see or touch them. So the idea behind this research was to do this kind of study in Lake Malawi for the first time. So for the objectives, we wanted to compare the species lists, comparing to the Troll survey that was conducted in 2023. This one was by the Department of Fisheries, and then we wanted to see the differences in what they actually catch, like the physical catches against what we find with molecular work. We also wanted to detect any rare species that could be missing if we're using the Troll survey in itself, but also we just wanted to see the resolution of the primer pairs that we're using, because we know that there is a lot of because of the vast radiation in Lake Malawi. So we were trying to see how far the resolution could go for us to see if we can use eDNA as a way of biomonitoring in the lake. So we had 2 sampling events. One of it happened all of it was on Lake Malawi, but all in all, we collected about 143 environmental water samples. And then they were kind of like throughout the whole lake, from Karonga to Chitipa. But also we had, So we had one across the whole lake. I hope you can see my pointer. And then we also had one only at the Lake Malawi National Park, which Dr. Katongo talked about at a certain point. So the park where there is all of this protection happening was kind of like another place that we also sampled during this time, but also the whole lake as a whole. For the lake, the Lake Malawi National Park, we collected the samples at 0, 20, and 40. So it was kind of like a vertical profiling to see if there's going to be differences in the samples that we see per depth, because the fishes of Lake Malawi are actually dependent on the depth itself. So we did a vertical profiling because there was no physical catches related to this one. But on the Lake Malawi, the whole lake, we collected at the depth at which the troll was. So it was hold. So if the troll hold at 100 meters, then we would collect water at that. For the data analysis and for the markers, we used 2 sets of primers. We had the mitochondrial primers, the universal ones, the MyFish. And then we also had the custom nuclear primers. This was because we wanted to see the resolution again, and we know that the mitochondrial DNA itself cannot help us with cichlids. So we tried we had our own custom nuclear primers. The deep shallow Utaka, we also had diplotaxodon as well as the Mbuna raphochromis. So these ones were all designed using 1,375 whole genome sequences, and it was assessed in silico using ASCAL 1.2 reference genome. And from there, we were able to see it worked. We tried it on RiverCam, but when we came to Lake Malawi, we were also trying to see if it's going to work on our samples in itself. Okay, I should also mention that because of how much our lake has got all this radiation and whatnot, we wanted to bring down the reference database that we mapped it to. We created a specific one for Lake Malawi fishes alone and any other life that you could find there. So we were mapping to a narrowed down kind of a reference reference to make it easier for us to map it. Looking at the 12S MyFish region, we were only able to split the cichlids from the non-cichlids, and but we could not actually see any difference between the cichlids as it was already expected. So because the MyFish, we were sure we were going to find this, it was expected, and it was in turn found that we could only separate or split the cichlids from the non-cichlids, but it was also strong enough to also detect the non-cichlids and separate them at species level as well. And also it was also able to kind of like help us to have an idea how far whether we can actually use whitefish for the cichlids or not. And we really it really proved that we couldn't use it as a whole. So for the sake of the two data sets, we had samples which were, okay, so all the training for sampling, library preparation, sequencing was all done in the University of Cambridge. So where you have TS-CAM, that means it's a troll survey data from the Cambridge dataset. So this one was all processed at the University of Cambridge. But then because we have got a lab that here at the University of Malawi, that has also the ISIC 100. We did everything that we did in University of Cambridge. We tried to replicate it and to do it again at the University of Malawi to see the differences between the two platforms. So obviously, Cambridge, we use the Sanger sequencing, we use the MySic, but for here, we use ISIC 100. So when you see TS ZOM, it just means University of Malawi Zomba, and then for the TS-CAM, it's all the data set from Cambridge. So this is only the troll survey. I will also look at the Lake Malawi National Parks study. So for the case of when we combined the TS, ZOM, and CAM, because they're all coming from the same, they're all from the troll survey comparison with eDNA, we collapsed all the pseudocranial all the cichlids. We tried to bring them together because of the huge mitonuclear discordance in Lake Malawi. So we collapsed them together because obviously we could not separate them by species level. So at the bottom here, the light blue could show you that there was a lot of cichlid DNA found, but this doesn't speak to individual species because we had to collapse it together. But at the same time, the non-cichlids, you were able to separate them. For example, one of the species that was very prominent was a Yuger lycopris, our common Usipa. And it was true as to how they are they're all over the lake and at different depths all over. So that was actually seen across all the sample states. And then the red that you see, red, pink, is all of the catfishes. So I have the Bagrass, the Claria, the Chilogladness, and we're also able to detect all of these ones. And then we could also find the Enteromyas at different places as well. So it kind of gave us a picture as to where a certain species could be clustered, but when we're looking at the non-cichlids. And this was in relation to what we found on the troll as well, which is in this slide. So when we try to compare the general presence and absence comparison of the eDNA data from the troll survey, we could find that, so the top half over here is all eDNA and the bottom half is from the catch. So you could find that, for example, Cynodontis was very prevalent and you could see it in the troll physically, but you could not actually detect it in the eDNA. In this case, it wasn't always the truth because during our sample analysis, during our analysis for the eDNA data, we had to cap at how many reads we could actually record to see to conclude as to how much DNA reads we should accept for us to plot. And then some of the sites were also removed for having some anomalies. So because of that, some of the samples could not be seen, but it was very true for all the cichlids in this case, and we're able to see that in as much as we're not able to actually or physically see the Aureochromis niloticus in the troll, but we're able to detect it in eDNA. So this was added into the reference panel because of the sighting in 2013 by Martin Jenner on the catchment area on the Tanzanian side of Lake Malawi. So we were also worried to see if there's going to be a relationship where we find the eDNA across the catchment area where the Tanzanian side is. But of course, we found it, KK is Nkhotakota, which is very far off from that place as well. And also in Salima, which is very far from the northern side where the Tanzanian side of the lake is. So overall, we are not alarmed by this, but it's a way for us to actually seek more molecular methods to detect some of these species because otherwise we're not going to be able to see them. At the same time, for the Momeridae, they are evasive species. Most of them are tiny, they're small, they're long, and they could escape the troll. So sometimes the troll would actually say, it would actually say there is no hominidae in that site, but with eDNA, we were able to catch it. So again, this brings to attention the importance of using eDNA against using only the traditional methods on their own. So for the national park data set, this was all processed at the University of Cambridge. And again, remember how we had this one with the vertical profiling at 0, 20, and 40. What was very interesting was the fact that at most of the replicates had, there were more fish that were detected at 40 meters compared to 20 meters. And at the same time, because this is a protected area, so we attributed all of this to be the fact that this place, there was a mix of all the species at different depths. So because it's a protected area, you are equally depths of the lake. We were also able to catch some non-fish DNA from here. So we found the bird, the cormorant, the cormorant. We also found the mammalia, which included the goat, the cow, and human DNA, which we attributed to runoff, as well as just some of it could be contamination during the whole process of handling DNA data. For the cichlid-specific premise sets, for all of them, because of the incomplete lineage sorting and all the integration between them, the performance of the cichlid-specific primers wasn't so good. So to a certain extent, you could see that the DSU and the MR, which is the Buna Raphochromis and the deep shallow Utaka, it performed kind of like okay, but when you look at the Dipodaxodon, it was very, very poor as to how much it could, the resolution of it overall. So, and all the readings that you can see in the Dipodaxodon graph at the top here, these are all coming from the Cambridge dataset and none of them from the ISIC 100 dataset. So we do not attribute the performance of these to actually the sequencing platform, but rather that we have to do more work on the primers themselves, because again, for cichlids, it's very, very difficult to separate them and to find because of the radiation. For the national park, again, this is what we saw, no Diplodocus reads. So in as much as this was also done on the Cambridge, with Cambridge on the MySic, we still could not detect Diplodocus. So this doesn't speak anything to the sequencing platform, but rather the design of the primer itself, but also how maybe impossible it would be for us to design nuclear primers for the cichlids, specific for the cichlids. As a summary, here is just how much the two methods fared. So for the eDNA, we were able to get a total of 13 families, and for the troll survey, about 8. For the genera, as you can see, 23 and 47. For the species, there were more species that were detected by the troll. But again, among the 171 that were detected by the troll survey, they were mostly cichlids. And because we could not separate the cichlids with our eDNA, this was kind of like an unfair comparison for us to do. But because we grouped all the Pseudocornylabrini as one group, so we took down the number of species detected. But when you look at the non-cichlid species, yes, and when you look at the 12S of the myfish, it could work and we could actually get the non-cichlid fish species properly. And of course, we weren't looking for the non-fish vertebrates, but being able to see the contamination there as well was also a good thing. And this gives us the thought that other living things can also be studied or biomonitored using molecular methods. So for the resolution, overall, it was just because of the Pseudocornylabrini and the Oreochromis being found across all sites, which is true. About the lake, we have our cichlids all over, and that the Claris and the Bagras were all over as well. We could find them in many places as well. This was also a good finding, because they are the least studied in the lake. So this could be a way of studying, like Hughes talked about this Anodontis njasei yesterday. It could be another way of actually doing biomonitoring of if they are non-cichlids, you could actually use these same primers to actually find them. To finish up, this is just the opportunities and challenges that are there with eDNA. But when we look at only the opportunities, we really have a great place to start from, knowing that we can actually do this on our lakes, or maybe in lakes that are not very cichlid rich, for example, or where we have got less number of species in itself. We could, instead of doing all the physical kind of monitoring, we could use eDNA, or maybe just use it as a complementary kind of a option. And the challenges, which the contamination sensitivity, which was a very good place for me to learn how to handle DNA samples at different stages of the whole process. So all in all, it was a success, and we have got very good results to say we can actually use this, but not maybe at the moment to use them on cichlids, but maybe design more primers or just use them on the non-cichlids. Many thanks should go to the University of Malawi and my supervisors, and University of Cambridge and Wellcome Sanger Institute, which was the funder of the whole project, and Hills Kaboa from the Department of Fisheries, as well as from the University of Malawi. Thank you.
I guess we missed you so much here in Eldoret. This subject on genomics of fish is so interesting. It is not just ending up in this conference room, it is going out there, and we're really enjoying the discussions. So thank you again. I think let's allow the interaction on your presentation. Any questions on Mandua's presentation?
Hello, I'm Olivia, and thank you for your presentation. I just have a question about your sampling method. From the maps you showed, it looked like you only sampled close to the shore and not in the middle of the lake. And then the first question would be, is that true, or are my eyes really bad? And if it's true, why did you make that decision? Is it only logistical? And would you expect like differences in the results if you would compare the shore and in the middle of the lake? Thank you.
Thank you, Bandua. You can respond.
So Olivia, what happened was it was an opportunistic sampling for me, because this was a 2023 biomass survey, and these stations were all designed by the Department of Fisheries. I had no control over which areas they were going to go to. So they always troll on the shoreline most of the times. The only part that we went that wasn't, it was also on the shoreline, but on the Mozambican side. So overall, it was all on the shore because of the vessel that I was using for my sampling. I had no choice. But further studies would be to sample, to go deeper into the lake. And that's the other beauty of eDNA, where you cannot sample using a trawl for whatever reason, because Lake Malawi is super deep as well. So where you cannot sample using the trawl, you can sample using eDNA, which is a very good thing that we learned from this as well, because you can actually go to all the places that are otherwise inaccessible. But for me, the design was on the shore because of where the boat was going. So logistically, I just jumped into doing, it was opportunistic to study the whole lake on my own would have maybe been difficult. But yes, it could be good to go deeper. At the same time, for our fishes, it happens that there isn't so much life in the middle. There is more on the shore. I believe it's because of the habitat selection of the fishes, the rocky areas which are mostly on the shoreline. So I'm also thinking that could be one reason it could be better to sample. It would be a more informative study on the shore than on deep inside, which I cannot speak to 100%, but I think that could be the case because most of our cichlids are rock dwellers, most of our fishes are rock dwellers, and most of our rocks are on the shoreline. But it could be a good opportunity to also go deeper into the lake and actually have a comparative kind of a thing to a comparative kind of a study to see what is happening between the two.
Yeah, I'm Ralph from Germany. Thanks for the very nice presentation, very comprehensive work. What I wonder, as you showed us with your general primer, you were not able to assign most of the samples to species level. And what make you choose this particular marker? I think it was the 16S, or what did you use? Or
12S.
Sorry?
12S.
Because I mean, like cytochrome B or so, they should go to the species level. Or were there other reasons why you didn't choose a marker which may have enabled assignment to species level? Thank you.
Okay. So for we also, yeah. We also used CO1, which I did not present here, but because we were trying to, one of the reasons we did this was to give the capacity to Malawi as well. So our sequencer is ISIC 100, and we cannot sequence CO1 on ISIC 100. So mostly because it's also the first study that we have had, and we just we know we can use MyFish, for example, on ISIC 100. That's why we picked that one, as well as the other nuclear-specific primers. So the choice was because of the sequencing platform.
Any other comment, questions? This is exciting. eDNA barcoding. So if there is none, I want to thank Mandua for that nice presentation. And next time, welcome to Kenya.
Thank you so much.
And we have one other session remaining this afternoon, so don't exit. Let us enjoy the discussion. So I think that closes our discussion on the theme on genomics on fish. And I want to thank the presenters for that very insightful engagement. When I look at my watch, I can't believe we are past 4:30. That tells you how enriching these particular presentations have been. So probably, I don't know, we need to get a way forward, whether we have a cup of tea, and then we come to for the next session. Yeah, we can grab a cup of tea for about 10 minutes, and then we come back for the next session. So karibu kwa
Nowadays, I'm more inclined towards immunogenetics, where we're doing HLA sequencing in the country as part of the project that involves other African countries. We want to understand the HLA in line with the new upcoming HIV vaccines. And we are also doing B-cell receptor sequencing, T-cell receptor sequencing. So basically, we want to understand how African populations can benefit from precision medicine, because precision medicine is the key thing. And like we have already seen that there's a lot of diversity in Africa. We have seen how fish is diverse, diversity in plants. It also happens that even in human genetic diversity, Africa is very rich in the same. So that means that most of the interventions that have been brought to us, they are really not tailored to us. So it is our honor to really understand our genetics such that we can be able to also align our medicines and vaccines with our population. So we are session 5 that from your program. This is a sub-theme on genomics and evolution of antibiotic resistance in agriculture, medicine, and environment. From your program, you can see that this session runs from now up to tomorrow, after just before tea break tomorrow. So we'll do the one half this evening, and then we'll do the second half tomorrow morning. So this session is quite short. We have 2 presenters that are lined for the presentation. We don't have a keynote speaker for this session, but tomorrow morning we'll be having our keynote speaker. So this evening is my pleasure to introduce Sylvia Chepkoskei. So Sylvia will present on whole genome sequencing of bacteria contaminants and AMR in Lake Victoria net tilapia supply chain. So Sylvia, you're welcome.
Thank you very much. As you've heard, my name is Sylvia Chepkoskei, an MSc student at the University of Eldoret, MSc Microbiology. So today I'll be presenting on whole genome sequencing for bacterial contaminant identification. I know in the abstract it also reads AMR. The AMR bit will be presented by my co-author, Dr. Salina Rono, who was my supervisor. Okay. We've heard so much today about aquaculture. And aquaculture is among the sectors that promote food security, generally in the world and here in Kenya, generating employment and improving household incomes. And Nile tilapia, which is Oreochromis niloticus, is a major fish species that is mostly farmed in Kenyan aquaculture and is a rich source of food fish in the Lake Victoria region. Therefore, microbial contamination poses a lot of risks to food security, and it occurs at any point that reduces the product quality, that is the fish product quality, threatening public health and causing vast economic losses. Fish is also a source of nutritive human food and economic benefit, as I've said. These also provide very quality rich proteins, as my other presenters have highlighted. And when fish is contaminated, it is a health hazard, which contributes highly to most of the foodborne illnesses due to consumption of contaminated fish. Fish is also perishable and highly susceptible to spoilage, leading to decreased quality of fish, mainly due to either handling, post-harvest losses, and other contamination. So the increased demand for consumption of fish, that is locally and in the export market, due to the increased demand of fish, it is therefore very key to understand the general public health risks that are associated with the consumption of fish throughout the entire supply chain. This also, the study on whole genome sequencing improves precision in bacterial contaminant identification due to its higher resolution, other than cultural techniques, biochemical tests, or basic PCR alone. This also strengthens traceability from the farm to the market and supports evidence-based food safety interventions. This study also provides data for surveillance, risk assessment on food quality, and policy development using a One Health approach. So the objective of this study is to characterize bacterial contaminants along the Lake Victoria Nile tilapia supply chain using whole genome sequencing. That is along the supply chain from the farm all the way to the retail markets. The potential contamination points include the production environments, that is the cage water, the sediments in the water. The feed can also introduce contamination, the handling equipment. At the harvesting stage, we have the nets, the crates, knives, and the workers that may potentially introduce contamination to the fish. During landing and transportation, the use of contaminated containers that are not handled properly, poor icing during transportation, and temperature abuse can also vastly lead to bacterial contamination of fish. At the wholesale and retail stages, we have the water that is used to wash fish. The processing surfaces can lead to contamination, cross-contamination of maybe the processed and the non-processed fish, and inadequate hygiene also contributes to bacterial contamination. For this study, the materials and methods. This study was carried out along the supply chain from cage farms in Lake Victoria, that's in Anyanga Beach, specifically in Usenge in Siaya County, all the way to Eldoret, Uasin Gishu County, the fish market. So at the farm level, it was at Siaya, that's Anyanga Beach, and the wholesale and retail points, as well as some of the transportation samples were taken from Uasin Gishu. This is the study sites, and as we can see from this picture, there are some of the unpleasant conditions that fish are subjected to. So this is just the methodology. The study design was cross-sectional, experimental study. For the sample determination, we used that formula. And this was systematic sampling used to select cages, and then now random sampling were used for sampling for the sample collection. Then after the statistical analysis using R. So during sample collection, a total of 179 samples were collected along the supply chain, and the samples were transported within 6 hours using iced transportation. Some of the samples that were collected included cage water, the fish swabs, swab of the boats, the processing surfaces, the processing water, swabs of containers, transportation vehicles, surfaces used for fish processing at wholesale, both at wholesale and retail points. This is just to gauge the level of contamination along the supply chain. These are the sampling protocols that was used. So for the morphological characteristics, identification of E. coli was done using this method, using the TBX medium by spreading 0.1 ml of the inoculum on the dried surface of that media, then followed by incubation at 44 degrees for 24 hours. The resulting colonies were blue-green colonies. These were the QC that was used, the positive and the negative controls. Salmonella was also identified by using buffered peptone water as pre-enrichment, and then the inoculum was put in RVS and MKTTN broth, followed by incubation at 41.5 degrees, at 41.5 for RVS, and 37 for MKTTN for 24 hours. Then streaking on XLD and hectoin enteric agar at 37 degrees for 24 hours. The presumptive colonies were black. And then biochemical confirmation were used. That is the microbact identification kit, 24E. These are just the organisms that were isolated at the lab. Staphylococcus aureus was also used on BP agar by spreading, then incubating on at 37 degrees for 48 hours. Black colonies with clear halo were presumptive colonies of Staphylococcus aureus, and then incubation in 37 degrees on brain heart infusion agar, then confirmation by microbact kit, 12S kit. These are the QC that were used. So Shigella was also identified using Shigella broth, and then containing a novobiocin, incubated at 41.5 degrees for 18 hours, and then spread on hectoin enteric agar, incubated again at 37 for 24 hours. Then blue-green colonies were confirmed by biochemical confirmation. So from the lab level, these were the data that were produced. We can see there that the highest abundance of bacterial species from the cultural characteristics was E. coli at 28.7%, followed by S. aureus, then Salmonella. And the distribution along the supply chain, we noticed that at the lake level, that's where most of the contaminants were, followed by the retail, and then the wholesale level. So the bacteria that were not able to be identified at the lab were sent for further identification using whole genome sequencing. At UK, a lab called Microbes NG, and this is just an illustration of the whole genome sequencing workflow that used Illumina-based platform using 2 times 250 base pair kits at a coverage of 30x. So the cell lysis was done using TE buffer and lysozyme and metapolyzyme and RNase A, followed by treatment with proteinase K. And then DNA quantification was done using quanti-IT, double-stranded DNA, HS assay. The genomic libraries were later prepared using the Nextera XT library preparation kit, and the purified DNA was subjected to the solid phase reversible immobilization, beads and suspension in elution buffer. Then from there, the Illumina short reads were sequenced, followed by bioinformatics analysis and phylogenetic tree construction. So the phylogenetic tree were constructed using the maximum likelihood method, constructed using IQ tree, and that's the model that was used, evaluated with 1,000 ultra-fast bootstrap replicates, and the resulting trees were visualized using Fig Tree version 1.44. And for the bioinformatics, the trimmed sequences were downloaded, and then the summary reports generated using multi-QC. The assemblies were in FASTA file Kraken 2 for the taxonomic classification, and the quality assessment was performed using FastQC. The de novo assemblies was done using SPADES. So these are some of the, I just sampled the top, I think, 23. And these generated a total of 3,360 microorganisms. So based on their relative abundance, I have arranged them according to their abundance. The most abundant was Staphylococcus ludinensis, followed by Staphylococcus epidermidis. So among the genus that were the most abundant was Staphylococci genus. So it's important to note that Staphylococcus ludinensis is not necessarily a pathogenic bacteria, but it can be opportunistic. It's an opportunistic pathogen, meaning if there is a lapse, if there is a lapse in food safety measures, these organisms can cause harm. Of course, some of the listed microorganisms here are pathogenic, like Salmonella, Staphylococcus aureus, which can cause diarrheal diseases, Escherichia coli. These are some of the organisms that came out from the results of the whole. So this is just an illustration of what I have just displayed of the species distribution across the samples along there. These are just some of them of the species that were present along the food chain. So we can see from the far left, FR means it is fish from the retail point, and all the way to we have for the retail we have the wholesale, and most of the most abundant species here we find them at the wholesale point. That it correlates. It almost correlates well with the findings from the lab cultures. So to conclude, so the most predominant bacteria was Staphylococci from the whole genome sequencing, E. coli, Staphylococcus aureus, Salmonella species, Shigella, and Pseudomonas putida, that were mainly detected at wholesale and retail points. So phylogenetic analysis also revealed several variants of the identified bacteria. I think the phylogenetic tree is coming after, sorry for the mix-up. So, and we find that whole genome sequencing is also a very powerful tool in identifying bacterial contaminants that the cultural characteristics may not bring out along the Nile tilapia supply chain, and as well it shows the genetic relatedness between the species, and therefore the application of these technologies in these supply chains can improve food safety measures and the strategies that are aimed at supply chain management. Sorry, I think I skipped the phylogenetic tree, my apologies. I acknowledge the University of Eldoret that they gave me an opportunity to study there. And the GCRF, because part of the work was funded by the GCRF through the academy, by the Academy of Medical Sciences through the GCRF project, and my employer, Kenya Bureau of Standards. So these are some of the pictures from Usenge. These are the cages that we took our samples from. And this is my team, my supervisors, and Dr. Ezra, who assisted me, and an undergraduate student called Faith. So from the last two pictures, we can see some of the conditions that fishers are subjected to. The crates are just placed on bare surfaces, which can pose a risk to public health, and as well as the processing surfaces may not be that adequate and suitable for food processing. Thank you.
A clap for Sylvia. Sylvia, thank you very, very much for your presentation. It's very insightful. So we welcome questions and comments from the audience.
Yeah, thanks for that presentation. I want to understand whether, because you will say you sampled along this supply chain, so is there along that supply chain now an kind of an accumulation of bacterial species so that we get more and more and more towards the end, or is it like that you pick up bacteria but they are replaced maybe by other bacteria in the next point? So how is that looking like along this supply chain?
Yeah, thank you for your question. Sorry, I think I skipped highlighting that. We noticed that as you go along the supply chain, the contamination increases vis-a-vis the ones at the lake, probably due to human intervention and contamination that are introduced by humans. Also, the water that is used for processing fish may not that be. We notice that most of the retailers and the wholesalers are recycling processing water, posing a risk again to the consumers.
We have another question.
Yeah, thank you, Sylvia, for the nice presentation. I have one question, and this might be in line with you said you're working with KEBS. Yeah, so in regard to, quality control on our food products, and we are seeing you use whole genome sequencing. So how applicable is that in the food control routine, quality control measures in terms of that, like in KEBS, or what could be the application of your methodology and the outcome, and how can we apply that? Yeah.
Thank you, Daktari. I think curbing this would involve a lot of multi-sectoral collaboration. That is the government, private sector, including the farmers, just to come together and see how best it can be done. But from the government side, I think this needs a lot of highlighting so that they see the need for incorporating such high throughput technologies to this kind of work in ensuring that public health safety is guaranteed. From a KEBS point of view, I don't know if I'll answer you well, because for us, we do regulate food that has been packaged, but there is a role that we can play, of course, because we develop standards. So we can champion the use of such technologies in our standards that can enhance testing in food safety. I don't know if I've answered your question.
Partly, but I'm just wondering in terms of cost effectiveness. So that's why I'm asking, when will we be doing all genome sequencing and trying to say we are using it in these measures of, quality control? So I'm not sure about.
Yeah, I know some of these technologies are quite expensive, but we are hoping that in the future we get to that level that we are able to incorporate such technologies, maybe through funding and other collaborations, just to ensure that this is done.
Thank you. I think there's more contribution in that line.
It's directly to that topic. So I don't believe that in the future, when you want to implement something, you need whole genome sequencing. So once you know what is there, so from there you can develop probably more cost-effective targeted assays that check for some of the relevant bacteria. There might be bacteria that are not really relevant. Is that right? So you said there are some opportunistic, but you also, there were many that you didn't mention that were probably less problematic, and we don't need to look for them, right?
Yeah, sure, that's good contribution. Thank you.
Thank you. Any more reactions? I feel this is a good topic because now you're in regulation. I think it's in line with what you want, because what the food that comes to us is of good quality, and KEBS has that mandate. So I think, as the contributors have said, I think now that you have used state-of-art tools to know that there is a problem, but the interventions could be more cost-friendly, such that people can get healthy foods in there. So do you think that you could influence maybe a policy that guides to maybe more food that is not contaminated, or fish that is not contaminated?
Yeah, sorry, I think I missed my slide on the recommendation. It was one of the aspects was we are hoping that such studies are able to influence policy in terms of yeah, I think I missed that point on that. One of the recommendation was to influence policy on such. And at the regulator's point of view, we have the opportunity that sometimes we sit in the technical committees meetings, so we are better placed to front such recommendations. Thank you.
Thank you. Any more reactions? So if not, thank you, Sylvia, for your presentation. We can give a hearty clap to Sylvia. So we have, I don't know if Vivian is in. So the other presenter, Bashir, I think was supposed to present online. In the absence of the then two presenters, I think these sessions come to an end. The other session will be there tomorrow morning. So I don't know if there are some final comments for the day. James?
Yeah, so thank you, Samuel. Yeah, not much of the comments, but just to thank you, all of you, for your patience and your active participation. So as we end, we just go into a session where you can network or interact until around 7. Then dinner will be available from 7 to 9 for all of us. So don't leave, because there's dinner for all. Thank you.
All right.